professor in Osmania University since 2014. A postdoctoral fellow. He was there as a postdoctoral fellow 2013 to 14 in Biomed Innovation Center, Heidelberg, Germany. He was also a postdoctoral fellow from 13 January to 10th January, University Medical Center, Heidelberg, Germany. Heidelberg, Germany. He was also a postdoctoral fellow from 2011, January 11 to 2012, December, Technical University of Munich, Germany. He was also there as a PhD student in the same university, Technical University of Munich, Germany. He was a junior research fellow, University of Hyderabad. He was a junior research fellow in the L.V. Prasad Eye Institute, Hyderabad, India. And also he was a student of Osmania University of Calicut, Kerala. He was there for some time as a project fellow, MSc dissertation for Center for DNA Fingerprinting and Diagnostics, CDFD in Hyderabad. He was a trainee as a MSc student. He gone through the summer training in the CRI Institute, Cancer Research Institute, Mumbai, India. In nutshell, very briefly, he has published several national and international publications. Altogether, it is 24 in number. Number is very small, but the quality is very high. I can say proudly. The aggregate impact factor is 140. Number of citations 600, and H index is 11. So it shows entire quality of Dr. Ramakrishna research work, research contribution. And scientific presentation, international 6 presentations, international poster presentations 20, and invited talks 10. And Sir also member of international bodies, several national, international and national bodies. Here, very few international bodies: American Association for Cancer Research, a full member; European Association for Cancer Research, ambassador member; and a national level, Telangana Academy of Sciences. He is associate fellow of Telangana Academy of Sciences and Indian Science Congress, a fellow. And awards, he received awards as a MSc student, a DBT from DBT, Department of Biotechnology, and a junior research fellow, CSR India, and a travel grant, ISSR travel grant for Nottingham, UK, and EMBO Welcome Trust, a DBT Welcome Trust Award to Barcelona, Spain, European Society of Endocrinology Travel Award to Odili in France. Presently, he received research grants from different agencies. Here, one is UGC FRP Startup Research Grant, 6 lakhs, and DBT SERB Extramural Research Grant, and it is around 49 lakhs. So it is very in brief about the Dr. Ramakrishna. He is very good friend of all of the organizers, and is a really good resource person, good resource to the upcoming technologies in especially plant sciences. I welcome you, sir, for the today afternoon session. The talk will be application of genetic techniques in molecular taxonomy. Definitely, I hope and assure all the audience that definitely you are going to enjoy this talk today afternoon. Thank you, sir. Thank you.
Thank you very much.
Dr. Ramakrishna to share the screen and continue the talk.
So I guess the screen is shared now, right?
Yeah, it is. Can you make it a full screen, sir?
Yeah, I will do. I will also exit my video so that we can concentrate more on the slides.
Yeah.
So thank you very much for giving me this opportunity. I thank my friends and the hosts, Dr. Naishwar Rao and Dr. Vijay Bhaskar Reddy, along with other co-hosts. It's my pleasure to talk to you people. So I was told that it's a mix of faculties, research scholars, and master students, especially with respect to botany. So I will limit my talk mostly to the basics of these various molecular biology techniques that are used for molecular taxonomy. I'm sure you must have received enough of the introduction regarding molecular taxonomy in previous sessions, so I also don't go much into it. I will just cover briefly about it. So the title, as you see, is Application of Genetic Techniques in Molecular Taxonomy. Basically, I'm a cancer biologist, but I am housed in Center for Plant Molecular Biology. The only exposure I had for plant biotechnology is during my MSc at University of Calicut. We were doing a course on plant genetic engineering, and we were doing that at Indian Institute of Spices Research, Calicut. So every weekend we used to travel there, had our theoretical as well as practical classes there. So I would say after a long time, this is an opportunity to talk about plants again. Coming back to the topic, what is molecular taxonomy? Taxonomy in general, as you know, deals with exploration, description, naming, and classification of organisms. And classical taxonomy is based on several features or characteristics that are ecological, morphological, or cytological, or sometimes biochemical in nature, which has its own limitations because many of these features are also influenced by the environment that may lead to inaccurate classification of the species you are studying. So most recently, the advent of molecular taxonomy, which depends on this classification based on more stable molecules such as DNA or RNA or proteins, which very much remain constant between individuals of species or subspecies. Just to go into a bit of history, as you know, classical taxonomy was based on morphological characteristics and so on, but molecular taxonomy initially started with the analysis of micromolecules such as molecules that are responsible for the scent, for color of flowers or sometimes leaves, and chemicals that are released by the plants for chemical defenses, etc. This is appropriately called as chemical taxonomy or chemotaxonomy because this relied mostly on the chemicals that are present in plants. If one has to take the examples, you can say alkaloids, flavonoids, anthocyanins, and so on, which are specific to certain families or members within the species or subfamilies. Then there is also classification based on macromolecules. I give here 4 examples. First one is isoenzyme variability and serology. Isoenzyme refers to enzymes which are proteins. Proteins, as you know, is one of the macromolecules that are present in the cell, others being carbohydrates, lipids, nucleic acids. So because of polymorphisms or mutations arising at genetic level, there are different isoforms of proteins, which you call them as isoenzymes, and they can be studied using various biochemical techniques, and you can isolate these isoenzymes from known or unknown species and compare them, whether they are resembling closely or they vary widely and so on. And serology is where you take the antibody test. The antibodies that react with certain proteins within a plant are tested for their reactivity with proteins isolated from the test plant. Then the second example is DNA hybridization. It is based on reannealing of DNA fragments that are melted at high temperature. So you take DNA of your reference and reference plant, or plant material. And there is some background noise, maybe if everyone can mute except me, then it will be clear. So, yeah. So in DNA hybridization, you take DNA from a reference plant and you also isolate DNA from the test plant material. Hello. Yeah, I think the others can mute so that it is not interfering with the lecture. There is a lot of background noise. So you take these two DNAs and then you melt them and try to reanneal them by decreasing the temperature. For example, if you are able to reanneal these two DNAs at higher temperature, that means these two plants are closely related. And if you need to go down to lower temperature, then they are like distantly related. Then the third method is restriction site analysis of chloroplast DNA. Chloroplast DNA is relatively very smaller compared to the genomic DNA. So you can digest with a non-restriction enzyme and compare the patterns of restriction fragments between the reference plant species and the test plants. And finally, the most recent one is DNA barcoding method, where you try to sequence certain known sequences either in the genomic DNA or in chloroplast or in the mitochondria. And you compare the sequences of these short stretches of DNA and check how closely or how distantly they are related and classify accordingly. And we deal with this part of phylogenetics in the end of this presentation. So we will discuss about certain methods that are used for molecular taxonomy. But before going there, we need to know certain basic molecular biology techniques that are used for the isolation of genetic material, that is genomic DNA or chloroplast DNA in most of the cases. Then we will also discuss about the technical polymerase chain reaction. So if someone is already well versed with this, pardon me, because I'm told that I have to deal with postgraduate students or research scholars of botany. So that's why I have to go to these basics so that they understand the later parts of the talk. So I talk about isolation of genomic DNA, then isolation of chloroplast DNA briefly, and then the polymerase chain reaction, a bit of restriction enzymes, and how to analyze all these, how to analyze the data using all these techniques. I will deal in the second half of the talk. So isolation of genomic DNA from plant material is a bit difficult when you compare to that of the animal tissues or cells, because as you know, they have a thick cell wall. Along with that, you have several contaminants such as polyphenolic compounds, pigments, and so on. That is why the genomic DNA isolation method differs a bit from isolation from animal material. So briefly, I give here an overview. The material which you use could be either root or parts of shoot, such as leaf and so on. If you take leaf material, you have to make it brittle by putting it in liquid nitrogen and then grind them thoroughly. And the most important thing here is the lysis buffer for cell lysis. In case of plants, uses two very important compounds. One is the detergent CTAB. CTAB stands for cetyl trimethyl ammonium bromide. It is a cationic detergent. What it does is it dissolves the cell wall and plasma membrane, and it also helps in the separation of proteins and polysaccharides from nucleic acids. And apart from that, we also use polyvinyl polypyrrolidone. What it does is it prevents the oxidation of samples called browning, because this will interfere with the quality of the nucleic acid that you isolate. So since you are isolating genomic DNA, nucleic acid extraction also gets you some RNA which needs to be cleared off. That is why you treat it with RNase and enzyme that degrades the RNA. In case of certain species such as recalcitrant species, you also add STS to add some more strength to the detergent part. And then you use phenol chloroform procedures also in some cases, because of which you get two separate phases. One is organic and one is inorganic. All the nonpolar compounds will get separated into the organic phase, that is phenol in this case. And your nucleic acid, that is DNA in this case, is trapped in the aqueous phase. And you also have a interface in between these two. And you then carefully separate the aqueous phase that contains nucleic acid. And this aqueous phase contains your nucleic acid, but you need to precipitate it for you to work further on. So you precipitate it in the presence of isopropanol or absolute ethanol, because of, and sometimes you also use salts such as sodium acetate or potassium acetate, monovalent salts. And once you precipitate the DNA, you remove the alcohol, and you also wash thoroughly to remove the remnants of alcohol, because that also interferes with the both with the stability of the nucleic acid as well as the downstream process such as PCR or whatever technique you are going to use. So this is one technique which is basically used as a starting material. And you also need to isolate chloroplasts in case you are looking for the genes that are present in the chloroplast genome. So here basically you have three different phases. I could see two participants raise hands. If we take questions now or in the end, it is up to the hosts.
Yeah, please, at the end, we'll take up questions.
Okay, perfect.
Please continue. Thank you.
So these three phases, three different steps include separation of chloroplasts, as you see in red, and then lysis of these chloroplasts to extract the contents, and then purification of the DNA. So this is one of the simplified protocols I picked from a paper published in PLOS ONE by Shi et al. If someone wants to have the detailed protocol, you can. It is a modified protocol of the existing ones. So all the boxes that are shaded in light green are like basic protocol, and the ones shaded in green are slightly modified ones. And these guys have optimized this protocol by incorporating their own steps, which are shown in yellow. But basically what they do in separation of chloroplasts is that they harvest fresh leaves and then homogenize them in buffer A. You can see the contents of buffer A on the right. And following filtration, they centrifuged to discard the cell debris and centrifuged once again and washed chloroplasts separately with buffer B, which basically ends the separation of chloroplasts. And used a lysis buffer. Here they named it as buffer C, which includes SDS and proteinase K. SDS basically denatures the proteins by disrupting the disulfide bonds, which are formed between cysteine residues. And proteinase K digests these proteins into smaller peptides. And once you have digested these or extracted the contents of chloroplast, you centrifuge them so that all the membrane and other debris is discarded. Now comes to the isolation of chloroplast DNA. cpDNA refers to chloroplast DNA, where you add phenyl chloroform isoamyl alcohol mixture, which is in the ratio of 25 to 24 to 1 part of isoamyl alcohol. Following that, all the remaining protein contamination is removed because DNA is complexed with lots of protein which need to be removed. And then you precipitate, like we have previously described, with isopropyl alcohol or cold absolute ethanol. And because ethanol is not, or isopropanol is not the good solvent for DNA, you remove them and you wash thoroughly your DNA pellet with water, and then you resuspend either it in clean water or TCDTA buffer. So this ends the isolation of nucleic acids either from the nucleus, that is genomic DNA, or chloroplast DNA. So another basic technique which is very important and which made our research easy is polymerase chain reaction. The material you get could be very scant for further analysis. Say, for example, if you are just taking little material from a herbarium, or maybe you are collecting very small amounts of pollen, or whatever scant material you saved, it could be very less. So the introduction of this PCR technique makes our job easier by amplifying the region of interest. What basically happens is, in the first step, it will denature your template, that is the nucleic acid you are giving as input that you have isolated from the whatever material you had before. And then you also supply certain primers that are specifically binding to your regions of interest. Of course, in techniques like RAPD, you supply the reaction mixture with generic primers that can bind randomly. But here we take example of specific primers that bind to defined region that is shown here. The primers are shown in yellow and pink. This step is called annealing because you lower the temperature following denaturation that is done at higher temperature between 95 degrees and 100 degrees. And most likely the annealing temperature is somewhere between 55 and 65 degrees of centigrade, depending on the length of the primer and the content of guans and cytosines in your oligonucleotides, that is primers. And once they anneal, you again increase the temperature, typically to 68 degrees or 72 degrees centigrade, depending on the polymerase you are going to use. So DNA polymerase is the enzyme that will extend the 3' ends of these primers so that you will get a newly synthesized strand that is shown as red on the left side and red on the bottom side also. This is the parent strand and this is the parent strand, and these are the primers that have annealed, and following annealing, this primer is extended, as you see here, as red, to synthesize a complementary strand to the template you have supplied. So you are getting exactly the copy of a template you have supplied with. So at the end of first round, you get 2 copies of it, and this is amplified exponentially, because each of these 2 copies will give rise to 2 copies each again in further rounds. And at the end of typically 30 or 35 cycles, you get quite a good amounts of DNA that can be used for downstream applications. And another important technique that is used for in molecular taxonomy is the tools based on restriction enzyme. We can discuss more about the techniques such as RFLP or AFLP later. But what a restriction enzyme does is it will recognize certain sequence in the DNA of interest. As you see here, there is an example of EcoR1. The site that EcoR1 recognizes, that is called EcoR1 restriction site, is GAATTC, as shown in orange here. This is the sequence the enzyme recognizes. This is palindromic in nature. That means you read in opposite side, it is same GAATTC again. And it cuts after guanine before adenine on both the strands. If you are reading it from 5' to 3' on top strand, or 5' to 3' on bottom strand also, giving rise to overhangs of certain nucleotides. We call such ends as sticky ends. These sticky ends can be used, for example, to clone this fragment into a vector of interest. For that, what you need to do is you also cut a vector with the same enzyme, so that the sticky ends of the so-called DNA of interest, you call it insert, is complementary to that of the vector, so that when you mix this insert with that of the vector, this DNA will go and bind there, and you can use other enzymes such as ligases to seal the nicks that are formed. So that is basically a principle of molecular cloning. So let's come back to our molecular taxonomy. So these are the techniques such as isolation of nucleic acid, PCR, and restriction digestions that are used to study the relatedness of different subspecies or species under study. So what are the methods you are using? So far we have discussed about tools. So the methods you are going to use to study is like certain methods are recommended depending upon the level of analysis you are going to perform. So there are 3 levels, whether you are studying relationship between the members of the species, you call intraspecific, or between species, that is interspecific, and between different genuses, that is intergeneric. So this is taken from the textbook Molecular Plant Taxonomy, Methods and Protocols. It's a very nice book. One can go back to it to refer what I'm talking about. So when it comes to intraspecific, there are basically microsatellite based techniques, SNP based techniques. Microsatellites are short sequence repeats. These techniques involve ISSR, sample, remap, IRAP, SAP, AFLP, and RAPD. SNPs stand for single nucleotide polymorphisms. So we will discuss in detail in coming slides. And for interspecific, you use technique that are utilizing either nuclear ribosomal DNA, which uses sequences such as internal transcript sequence, intergenic sequences, or non-transcript spacers, and so on. Or when you use chloroplast DNA, you use intergeneric regions for the study. And for intergeneric, you use either nuclear ribosomal DNA, for which you choose 5S or 18S or 5.8S rRNA. So we will see exactly what they are and where they are located when we deal both with ITS, IGS, and so on. And ribosomal DNA, and several of the genes when you are studying chloroplast DNA, which includes Rubisco, that is RBCL, that encodes for larger subunit of this enzyme, or mature SK, call it MATK, RPSAB, PSBB, and so on. And less widely used is mitochondrial DNA based method. So in case someone is interested in using mitochondrial DNA, so you need to isolate mitochondria first and then isolate DNA from mitochondria for your analysis, the same way as we have seen for chloroplasts. So let's get into details. I'm not going in depth, or I'm not showing results as examples. So when it comes to microsatellites also are called short tandem repeats or simple sequence repeats. They're a tract of repetitive DNA, either 2 or 4 or 6 nucleotides are base pairs long. Typically, sometimes you will also see repeats of higher numbers like 8 or 10 nucleotides too. And they are present in tandem next to each other, and they are called repeats. So you have a motif that is 2 to 6 base pairs long, and this motif is repeated several times. This could be 5 to 52, sometimes 100 to 200 also, as you see in some diseases. Okay, so these are typically present in non-coding regions of the genomic DNA, and they serve as markers. So what exactly are markers? Markers are those regions within the DNA that serve as landmarks to associate our phenotype in question or our trait under study. So if a phenotype is segregating in your study and you want to exactly find out what is the gene that is responsible for this phenotype, so you normally employ certain markers which are distributed throughout the genome and try to associate the segregation of these markers with that of the phenotype. Because they are distributed throughout the genome mostly in an unbiased manner, you normally narrow down to few tens of markers that are segregating with a phenotype or a trait or a disease. And then you do fine mapping again using additional markers, and then try to find out the gene that is responsible for the phenotype under study. So that is how you do positional cloning for a trait of interest. So microsatellite is one such marker. The name comes after comparison with mini satellites, which are slightly longer stretches of nucleotides with fewer repeats. Microsatellites are shorter stretches with more number of repeats. That's the only difference between mini satellites and microsatellites. Here I just put an imaginary example. An example consisting of 3 alleles, 1, 2, and 3 shown in blue, red, and green. N refers to certain number of repeats of a dinucleotide C and A. So the repeat for allele A is shorter compared to that of allele 2, which is slightly longer than the 1, and allele 3 has more repeats than the other 2. So when you amplify, we have just discussed, right? You are isolating the DNA, could be genomic or chloroplast DNA. And then you perform PCR on this with primers that are defined on either side of this marker, for example. And when you amplify this, I just skipped a technique called agarose gel electrophoresis. It is a technique which looks something like this. It is made of a polymer called agarose, and you have several wells where you can load your DNA sample and apply electric field so that the DNA runs in this direction according to the size. And you can see the DNA as bands, like here, not in so many colors, but typically in orange, because you use a dye called ethidium bromide that intercalates DNA. And when this gel is put on a UV lamp, the DNA is lit and an orange color is shown. So you can see DNA as a band, orange color band, when put on a transilluminator based on UV lamp. So what you do is you perform PCR, for example, on different samples, and you don't know which allele is present in your species of interest, right? So you take several samples and you run them on an agarose gel. So first you perform PCR, so these are amplified. If, for example, in lane 1, you have a plant which is homozygous for the allele 1, you have a shorter stretch of blue, right? And we are talking it's homozygous. So you have 2 copies of it at this locus. So you get a single band of smaller size because the repeats are fewer in number. So in the second example, for example, it is homozygous for allele 2, which is slightly bigger than that of the allele 1 because of more number of repeats. You get a band of higher size compared to that of the blue one. Similarly, when you have homozygous allele 3 at that particular locus, you get a even higher band, which is green in color. So you have other combinations too. If you have a heterozygote of first and second allele, you will get 2 bands because you have 2 alleles at this locus that matches to blue and red. So you get 2 bands, one matching the size of the blue and another one that matches the size of the red. So from this band pattern, you can say that it is heterozygous for alleles 1 and 2. Similarly, sorry, it is a bit distorted. It won't run that way. It should be straight just below this. So it is heterozygous for alleles 1 and 3. So you get a blue band for which is lesser in size and a green one which is highest in size. And you have a third combination that is heterozygous for second and third. And you get a intermediate size band of red and a higher size of green. So this is how when you use microsatellite markers that are at a defined loci, you can genotype your plants. And sometimes it is advised when you use microsatellite markers, you use radioactively labeled reactions, because when you amplify these dinucleotides, there is replication slippage. So sometimes some nucleotides are skipped while the polymerase is extending the primer. So you get additional bands which are not exactly of this size, but could be slightly lower in size. But they could be minor bands. You call them stutters because this happens because of replication slippage. So when you use a radioactive PCR, you could see all kinds of bands, not just the major band which you are interested in, but also stutters. And you can run them on long polyacrylamide gels and score the bands and genotype to find out what allele is present in your plant of interest. That is one technique. And what are the techniques based on microsatellite markers? The first one is called intersimple sequence repeat, ISSR. So this ISSR is the genomic region that is present between 2 microsatellites. See, as we have discussed, microsatellites are distributed throughout the genome, right? And when you take 2 different microsatellites, the region between them is called ISSR. And as with the microsatellite markers, ISSRs are also randomly distributed throughout the genome. And the length typically is between 100 and 3000 base pairs, depending on how closely or distantly these microsatellites are present. And you can generate 10 to 16 fragments simultaneously using a single PCR when you are scoring for ISSR. So you can understand here what I mean. So how do you design primers for ISSR polymorphism? Here is an imaginary gel. Here is the DNA sequence that you are interested in. For example, this is the one which is shaded in yellow is a microsatellite marker. This is microsatellite marker 2. If, for example, your primer is designed within the microsatellite markers, say for example this blue one, right? This is within the shaded region. It is not extending outside or inside. Same is the reverse primer. So you get several bands here. It is not just one band, because this could be variable, or this reverse primer could also bind to another microsatellite marker which is far away, and that will amplify a marker of larger size, and they could also be amplified at totally different regions. So you get markers of different sizes, but you can also specifically design your primer such that it partly binds to microsatellite marker and the other part either outside as you see in the pink or inside as you see in green. See here the pink primer starts 3 or 4 nucleotides outside the microsatellite marker, so that this PCR product is specific to this particular locus. It won't randomly bind to any microsatellite marker. Similarly, this primer starts binding within the microsatellite marker, but it extends into the ISSR region, so that this green one also results in a specific band. Both pink and green results in bands of specific size. Don't worry, there is no such a size difference between the pink one and the green one. Normally, this is just few nucleotides long, so the right depiction could be green also somewhere here. So if someone got that doubt. So you can specifically perform genotyping for specific ISSR at a particular locus by designing primers either pink or green, that means that extend either outside or inside the ISSR region. So coming to the next technique, sample remap and SSAP. Sample stands for selective amplification of microsatellite polymorphic loci. So sorry, I will just add one more point in the previous slide. So what we are scoring here is, once you get these products, it could be that there are certain insertions or deletions that could result in the changes in the sizes, or you can sequence them for differences in the nucleotide sequences. So in selective amplification of microsatellite polymorphic loci, like I have discussed in the previous slide, so you are using an AFLP primer. AFLP I will be discussing soon, which involves restriction dilution by an enzyme and then addition of adapters and so on. So one primer is AFLP primer, another one binds to a microsatellite marker. SSAP means sequence specific amplified polymorphism, where here one primer is binding the retroposon containing fragment, because you are choosing a region that is near the retrotransposon, and the other one binds to the nearby restriction endonuclease site. Remap stands for retrotransposon microsatellite amplified polymorphism. Here you are choosing a region that is present between retrotransposons and microsatellite markers, which is amplified by PCR that utilizes primers that specifically bind to LTR part of retrotransposon and within the microsatellite marker. And if you want to make it locus specific, you design primer such that that extends beyond or before the microsatellite sequence. So this picture will give you a clear idea of the difference between sample, SSAP, and remap. In case of sample, you have a primer that is complementary to AFLP adapter, that is a forward primer, and the reverse primer is binding within the microsatellite marker, which contains several repeats that are shown in shorter boxes here. And I don't have space to cite the reference. It is from the same book, in case someone want to refer to. In case of SSAP, the primer 1 is same that of sample, but the reverse primer, while in sample it is binding to microsatellite marker, in SSAP it is binding to the LTR element of the retrotransposon. This part is a retrotransposon, and this reverse primer is going outside the retrotransposon into the or towards the locus of interest. And in remap, so the primer 1 binds to the microsatellite marker. That is the difference between SSAP and the remap. In case of sample and SSAP, the forward primer binds to the AFLP fragment, whereas in case of remap, it binds to a microsatellite marker, whereas the reverse primer, like that of SSAP, binds to the LTR of the retrotransposon, also going outside the retrotransposon. So that is the difference between sample, SSAP, and remap. Then the technique called IRAP is there, that is inter-retrotransposon amplified polymorphism. What it does is the region between two retrotransposons is amplified by PCR using two primers that are running out of the LTR sequence. I will show the picture, then it will be more clearer. This is one retrotransposon and this is second retrotransposon, and the region between these two is inter-retrotransposon region, and this region is amplified. That's why this technique becomes inter-retrotransposon amplified polymorphism. And you see forward primer as shown in yellow, that is running out of retrotransposon 1, and red primer as you see here is also running out of retrotransposon 2. Both these amplify this region that is red in between, giving rise to the PCR product that can be analyzed. And if the retrotransposon directionality is different, you take this yellow primer instead of red there, and the idea is same. You are amplifying the inter-retrotransposon region, which will be used for analyzing the relationship between species. And then AFLP is one of the older techniques. It stands for amplified fragment length polymorphism. What do you mean by length polymorphism is that when you are amplifying a fragment that is defined, and if there is difference in sizes that alters the length of this region, you call it a length polymorphism, because there are differences in lengths between different alleles within a population. So when you are able to study that, the technique is called AFLP, or amplified fragment length polymorphism. So you perform PCR with primers. Sometimes they could be defined, or sometimes they could be very specific. And then you perform PCR and then you analyze on the gel. Another older technique is called random amplification of polymorphic DNA, RAPD. The only difference here is that the primers are arbitrary, they are not very defined. Sequence of about 10 nucleotides in length, and you will get several fragments which can be run on a gel and tested for the relationship between plants under study. So the final one, apart from microsatellite based marker for intraspecific studies, is SNP, stands for single nucleotide polymorphism. Here polymorphism means a nucleotide that is present at particular locus is just changed to another nucleotide, and this could be studied either by DNA sequencing, because that is the only way you can see the difference between fragments under study, or high throughput studies such as microarrays can be designed so that the hybridization is very robust enough so that they can differentiate between SNPs under study, and they are more in number, ranging from thousands to millions depending on the size of the genome, and they are uniformly distributed throughout the genome, and they are very useful for high throughput studies. So for interspecific studies using nuclear ribosomal DNA, you have 3 regions, that is ITS, IGS, and NTS. Let's look at them. ITS stands for internal transcribed spacer, which is present within the genes that are encoding for rRNAs that are specific for large subunit ribosome. I will show you the slide which will make it more clearer. IGS stands for intergenic spacer. ITS is present on either side of the 5.8 sRNA within the region that contains these 3 of the genes, whereas IGS is present between copies of these ribosomal DNAs. That is why it is called intergenic spacer. And within this intergenic spacer, there are also regions called non-transcribed spacer, which is upstream of the first gene of the ribosomal RNA. It will be more clearer now. This is the locus. This is one rDNA, and this is the second rDNA that follows this. So one rDNA codes for 3 rRNAs, 18S rRNA, 5.8S rRNA as you see in red, and 25S as you see it in gray. So the region between 18S rRNA and 5.8S rRNA is called ITS1, internal transcribed spacer, and between 5.8S and 25S is called ITS2, that is internal transcribed spacer 2. So both ITS1 and ITS2 are present within the rDNA that codes for 3 rRNAs that are transcribed as one transcript. This is one thing you need to remember. And the second transcript starts here and codes for again the same thing which runs there. So these 2 will serve as regions to study variability between subspecies or species. And the region between different genes that codes for this transcript and another transcript is called IGS. That is why it is called intergenic. This is one gene and this is another gene. And this is more variable. And within this is present upstream region. See, this variable region is amplified here. What all it contains is an upstream region, which is called non-transcribed spacer, and a downstream region from where, after which the transcription of the second rDNA gene starts. So this is with respect to the study of nuclear ribosomal DNA. So the genes that are normally used for study of phylogenetic relationships when it comes to chloroplast gene, the popular one is called RBCL. It codes for the larger subunit of the rubisco. It is nearly 1.5 kb in length. And it is pretty much conserved throughout the evolution. And it stands for ribulose 1,5-bisphosphate carboxylase oxygenase enzyme, because it performs both functions of carboxylation and oxygenation also. And this is sequenced, and the sequences are compared for phylogenetic relationship, and sometimes additional marker called MATK, which stands for maturase, is also used for genotyping. So when it comes to DNA barcoding, using several techniques, are basically more straightforward one recently is you sequence your plant of interest as well as reference ones and compare their sequences. This is what is called multiple sequence alignment. And totally black means the sequence is, the nucleotide at these regions is totally conserved, and their gray means that they are mostly conserved, and sometimes you see that they are not at all conserved with gaps. And using these sequences, you also can construct a phylogenetic tree. You call it reconstruction of phylogenetic tree, and based on their relationship, you get either a rooted tree or an unrooted tree. And what you can see is S1 and S2 are closely related, and S4 is distantly related from all these three. Here, the length of the line represents the distance of the relationship from the other three. So what exactly is multiple sequence alignment? It is alignment of many biological sequences. It could be amino acid sequence or nucleotide sequence, depending on what your input is, whether it is protein or nucleic acid. And using that sequence, you reconstruct a phylogenetic tree that visibly shows the relationship between different species under study. Recently, flow cytometry is also used to study these relationships. What you normally do is you take the plant material in isolation buffer and you isolate nuclei. Then you filter to remove all the debris, and you add a dye called propydium iodide, which will go and bind to nucleus, so that when you run in a flow cytometer, when a laser is exciting it, it will emit, and the amount of propydium iodide reflects the ploidy of the nucleus that is under study. That means if you are taking nuclei from pollen or egg, you have 1C. So the amount of propydium iodide you detect is of certain amount. But following fertilization, if you are taking from S phase or M phase, you get double the amount. And if you are taking from G2, or if you are taking some nuclei which have undergone endopolyploid cycles, you have more amount of signal that is coming from propydium iodide, which in turn is bound to DNA, which reflects the ploidy of the nuclei under study. And that is how you can, each plant has a defined or certain amount of DNA material, and when you are comparing the DNA of your test material with that of that, you can relate how close or how far is your plant from the test material. So I think basically I have briefly covered the overview of several techniques that are used for molecular taxonomic studies. And I think I have taken a bit more time because we lost some time during introduction session. And I thank you all for your patience. Over to the hosts.
Yeah, thank you, Ramakrishna. It's a wonderful lecture. And you have briefed everything about all the techniques used in plant taxonomy. And there are some participants who would like to interact with you.
Yeah.
Then over to Karthik. Karthik, are you there?
Yeah, yes, sir. Yes, sir.
Yeah, please.
1 second.
Please.
1 second, sir. So one of them posted about that one question, sir. Sir, in ISSR technique, is it always necessary to use micro or macro satellites?
Oh, you are. Yes, I will just go to the slide.
And
Yeah.
please turn on your video also, possible, Dr. Ramakrishna.
Yeah. Moment. This problem. So normally you use these primers which are binding within the microsatellites. Micro. And if you are really interested in studying specific locus, because when you are using these primers that are shown in blue, you are getting multiple fragments because these microsatellites are present everywhere, right? Still, that will give a fair amount of information. But if you want to study specific locus, it's better to use the primer that is also running either side of the microsatellite marker, so that you will get one discrete band. Yeah, next question. Hello.
Yes, sir.
Yeah, next question.
Only one question, sir. Someone asked. If anyone is having, please raise the hand. Yeah, Sri Lakshmi. 1 second, sir.
Mm-hmm.
One second, sir.
Yeah, sure.
Hello. Good afternoon, sir.
Good afternoon.
It was a very informative session. Thank you very much, sir. And sir, after seeing all your primer designing and the markers which you have suggested, I would like you to kindly suggest for any updated softwares to design a primer, which will be very useful for the upcoming research scholars in the plant
Sure.
field.
Yeah.
This I am asking because in the universities, the software availability is not updated. I mean, it may not be in reach to the students. So I want all the students to be well aware of the updated softwares.
I understand. So we
Yes,
have
sir.
a very important concern, especially people coming from non-knowledge technology background. So there
Yes, sir.
are lots of softwares where you can input a sequence of interest. So I will take two examples. Okay. First example is you have already decided which markers you want to do. So then you know what is the sequence of this marker which is available from public databases. So you can put this sequence of interest in some freely available ones, which I can share with you. I don't have at the moment right with me, but I can share with the host so that they can share with you also. So
I think that will be of great help, sir.
yeah. So then you can design, or the software will help you designing primers that are appropriate for PCR conditions also. And second thing, if you are just going with a totally new species and you don't know what the sequences are, there are certain wonderful databases which according to the species under question, they will suggest you markers and they will also suggest the primers to use. I will also try to share those resources with hosts.
Thank you, sir. Yeah.
Thank you.
Thank you, sir. One more question, sir.
Yeah, please.
Yeah, that's all, sir. Only two questions.
Yeah, one question is there. Jayati Biswas.
Mm-hmm.
In ISSR technique, is it always necessary to use micro or macro satellites?
Yeah, we always use microsatellites for such studies. So hope that answers the question.
This question is answered, sir, just now.
Okay. Yeah.
So I thank all the organizers for giving me the opportunity. So
Thank you so much, Ramakrishna. Ramakrishna is a very good teacher, very good researcher, and of course, a very good friend of me. And
very kind of you.
Ramakrishna, sir.
Sir.
Final one question, sir. Someone posted just now.
Yeah, please.
Sir, kindly suggest I'm using ITS1 and TRN L/F region for a gymnosperm. Is it good for it?
I can't exactly answer that now. If they can mail me, I can try to answer. So,
yeah, okay,
because I am not in this, I can talk to the guys who are available and I can definitely come back.
Sure. Yeah, one more question, sir. Can I have the author for the book for plant taxonomy methods and protocol?
Plant. Oh, okay. Let me check if this computer is open. I can tell just now. Or I will share with you, in case you are, okay, I could open it now. I will just tell the authors. Hopefully I'm not eating into the time of the next speaker. So the authors are John Walker from Hertfordshire, UK. John
Okay.
M. Walker. Okay.
Okay. Please, if anyone is having doubts, please raise your hand. And molecular taxonomy.
Okay.
Yeah, thank you, sir.
Yeah.
Thank you.
Dr. Nageshwara?
Yeah,
Dr. Nageshwara.
yeah. Thank you very much, Dr. Ramakrishna, sir.
It's my pleasure.
It was a valuable and wonderful lecture this afternoon. Definitely, if anybody interested, we'll make a mail to you. Please try to answer appropriately if possible from your side.
Definitely.
Thank you very much once again.
Thank you.
And Mr. Nagesh is ready to propose vote of thanks.
Okay.
Over to Mr. Nagesh. Nagesh? Yeah.
Sir, you cannot start your video because the host has.
No problem.
Good afternoon, everyone. This is Nagesh, Department of Botany, Nizam College. It's a great privilege to say vote of thanks for the afternoon session on application of genetic techniques in molecular taxonomy, delivered by Dr. Ramakrishna, sir. Sir, you have delivered a wonderful lecture, sir, on the various techniques of genetics and which are useful in the construction of phylogenetic tree. This session is really useful to our participants, sir. Thanks for your presence in this 1 week short-term course, sir. As well as I would like to thank our organizers, Vijay Bhaskar Reddy sir, Nageshwar Rao sir, as well as Shashikala madam for organizing this wonderful short-term course. And finally, I would like to thank our participants for their continuous active presence for this short-term course. Finally, I am very much thankful to the everyone for giving this wonderful opportunity. Thank you very much.
Thank you, Nagesh.
Thank you, sir. Thank you all.
Sir.
Madam,
Yeah, I want to speak few words about
madam, please.
Ramakrishna. Sir.
Oh, Shashikala madam. Yeah.
Sir. Good afternoon, sir.
Good afternoon.
A very great lecture we had now, sir. And apart from this, I would like to say one word, sir. Our Usmania University is great enough to hold such FRPs in our university, sir.
Oh, thank you.
Because I think your number is increasing day by day. Now your number is, I think, 6 or 5. I don't know exactly. I know one from chemistry and one from engineering faculty, geochemistry and biochemistry, and now you. It is great privilege to you people having here, sir. Thank you very much, sir.
It's pleasure working with you all.
Yeah, it's a great asset.
Because it is asset to our university, sir. Because FRP is, I think, it is a good honor to us, sir. Thank you very much.
It's our pleasure to work with nice colleagues like you.
Yeah, thank you, sir. Thank you, sir. In future, definitely we will try to be in collaborative work with you, sir.
Sure, definitely.
Thank you, sir. Thank you very much, sir.
Thank you all. Thank you.
Thank you, Dr. Ramakrishna. Bye.
Thank you, sir. Thank you very much.
And over to Dr. Nageshwara Amanji.
Yeah, thank you, Ramakrishna sir. Now we will start with the next session, next lecture. This afternoon we have a wonderful, one more young, talented speaker. Young and talented speaker today afternoon. That is Dr. Upender, Gangadharani Upender. He will be speaking on the Raman spectroscopy and its applications in life sciences, especially botany and zoology areas. It is my privilege, honor to introduce my young colleague, in the Osmania University. Dr. Upender, Dr. Upender Gangadharani.
Excuse me, Dr.
Yes, sir.
Upender, please turn on your video. So that,
No, sir, it's host is not allowing, it seems. You can't start your video because the host has stopped it, it's saying.
No, sir, it's allowed actually. Just take it out.
Yeah. No. No, still it is showing I cannot access the video.
Yeah.
I can't enable the video, sir.
Yeah. You will electrify, sir. One. Wait.
please continue.
Yeah, sure.
It is the case with me also. It's not allowing. Okay, no issue.
Fine.
I'll introduce, sir. Okay. Yeah. And Dr. Upender received his PhD from the Osmania University in the year 2010 and a master's degree from Osmania University in the 2003. In the year 2003, and he was awarded with the distinctions and various research awards. He got the distinction in bachelor degree, and he was the second topper in the masters in Osmania University. He was awarded summer research fellow, a summer training fellow, a fellowship from the Physical Research Laboratories, Ahmedabad, in the year 2002, and also he is UGC awarded a research fellowship in science for the meritorious students, JRF and SRF, in the August 2007 to 2010. He is also a fellow of UGC, Dr. D.S. Kotari, postdoctoral research fellow, and worked at University of Hyderabad, Hyderabad, Telangana. And he has vast experience in research, about 15 years. He was now presently is working as an assistant professor in Osmania University in the Department of Physics. He was a scientist C in CSIR, CRI Kolkata in the year 2000, March 2012 to March 2013, September. He was a fellow of Dr. Kotari Fellowship, a fellow of the Dr. D.S. Kotari in the University of Hyderabad. He was a fellow of a research scholar in the Osmania University from 2005 to 2010. It is very briefly about sir. And coming to the actual research input, he has published several national and international papers. Put together 35 research publications are there. In international 32 and national level 3, and the total number of citations is 532, and the average citations is per paper 9, and finally the H index is 16. So it is a good research contribution, very valuable contribution from the physics to the physics from the Dr. Pender sir. And he has collaborated and visited several national and international institutions. He is there with the Department of Atomic Energy, CSIR, Indore, and he is there with the people in collaboration with the ISCT and CCMB and NGRI, Hyderabad, and the University of Hyderabad, and also JNTU, Jagitchal in Telangana. And he has presented national and international conferences, 20 oral and poster presentations, and there are several lists there. I'll share. He is a referee or reviewer in many national and international papers, say a Journal of Alloys and Compounds, which are having each and every paper is having not less than 1 impact factor. So he will be a very good speaker in this beautiful afternoon, definitely. All the participants will enjoy the lecture also. With this short introduction, I welcome Dr. Upender.
Very good afternoon. First things first, let me thank Dr. Vijay Bhaskar sir and Dr. Raman Nageswara sir for giving me this opportunity to share some of the ideas relevant to Raman spectroscopy, and which is in particular related to the plant taxonomy or plant science. Very good afternoon, good afternoon to participants. To me, it is showing 128 participants. and probably some of them are teachers, some of them are researchers, some of them are young students. And of course, I have nothing to do with the taxonomy today in the afternoon, but mostly my talk related to the plant science, how Raman spectroscopy can be useful to characterize the plant tissues and related stuff. I hope you all find this talk going to be very useful in your research career in days to come. Yeah, Mr. Karthik, can I share the screen?
Yes, sir. You can share your screen,
Yeah,
sir.
sure. Is it okay? Is it visible now?
Visible, sir.
Thank you. You're right. Today, I would like to take you a very brief introduction on Raman spectroscopy, and I would like to focus some time on applications in plant taxonomy, how the Raman spectroscopy going to help us to characterize the plant tissues and that relevant stuff. As a speaker already introduced about me, and even though it is my customary to introduce myself to all the participants on this occasion. And my alma mater is Usmania University, and way back 2003, I studied my master's in physics discipline, and then I did my PhD from the same institution, and then I moved to my Kotari postdoctoral fellowship, University of Hyderabad. Then I was happened to be a scientist C at a CSIR institution in Kolkata. And probably that forced me or that compelled me to learn more about Raman spectroscopy. And I'll make you realize what is impact of Calcutta on Indian science. And of course, after a brief stay at Calcutta, then I moved to Hyderabad again, since it is my native place. So I found one job in university as a assistant professor. Then I left the Calcutta and joined Hyderabad and Usmania University. This is my brief details about alma mater. And coming to why you must show some intention or interest, or you have to divert your focus, since of course more often every day I'm visiting this particular platform, and I'm also keen showing interest to learn what other speakers are talking about. And to my surprise and to my knowledge is nothing to do with the knowledge that I've gained so far in the physics, because it's all botanists and plant scientists. They're all renowned faculty members who've been sharing the classification of the plants, and then different characterization tools and plant science and life sciences too. And what I can, in the beginning, and even I insisted, doctor, when he asked me, of course, my inclusion into this sort of platform is random or rather arbitrary. Since, I am basically physicist, I'm more into a material science or material scientist. What a material scientist can do in a plant science. A material, of course, a plant is also a material, but how my expertise I can bring into make you aware about Raman spectroscopy, and what the Raman spectroscopy can really do a job in plant taxonomy. And later, of course, he gave me some sort of time, and 4 or 5 days I had left over when he called me and asked my permission to inclusion in this sort of platform. Then I started searching the literature, and I was really astonished. Raman spectroscopy is playing a tremendous and very paramount role in plant taxonomy and plant science. And I don't know how many of you have been using the Raman spectroscopy into your scientific endeavors or in your research career. And later I realized, of course, instead of sharing my physics-based stuff, after I've gone through the literature, I felt Raman spectroscopy, which is very relevant tool and well above all the other, what do you say, microscopic technologies or microscopic techniques that have been implementing in plant tissues and plant science. And it is one of the superior techniques. And I make you realize when I go to the end of my talk, I bring you one important application of Raman spectroscopy into plant science. And I'm sure you will appreciate me, and you will also appreciate the Raman, how this Raman spectroscopy has turned as a universal and versatile tool can be applied across the scientific disciplines. And here I posed one question here. So why Raman spectroscopy is needed in plant science? What Raman spectroscopy can do in plant science? So here, since here I've mentioned few points, and the first important point, because it was the hope as a science graduate, as a science, what do you say, a relevant person, I hope you heard about Raman spectroscopy, and it was invented by great Sir C.V. Raman in 1928. It's almost 90 years old technique, and though it is 90 years old technique, still it's afresh. Why? Look at the first time the plant scientists applied the Raman scattering or Raman spectroscopy as a characterizing tool in the late 1980s or mid of 1980s. So I can treat is that 1990 probably science has began, or the people made use of the Raman spectroscopy as a scientific tool to imply its scope and applicability in plant science in the early of the 1990 year. Then since then you just count, it's hardly 30 years old. 30 years old is not that great age to understand the technique and to implement the technique and to exploit the technique to retrieve the required information, because it takes a lot many years when one particular what do you say, scientific effect or scientific phenomena came into science. It takes a lot many years. And for instance, Raman spectroscopy, if you take Raman spectroscopy 1928 in Calcutta by Raman and his student, okay, Krishnan, invented it. And since the discovery of lasers, hope you heard about lasers, being a life scientist, and you must be aware of laser and what does it do in the science. So 1960, 1970, up to that time, there was no laser. So almost since its discovery in 1920, Raman effect hardly didn't find any applicative role in the science to play. Okay, I'll come the details, the fullest details in the later slides. So reason why, it's a very new technique which any life scientist, life science student can make use of and can retrieve the detailed information related to the life science related stuff. And second point, if you look at and blue color font that I mentioned here, if you extract any sort of plant tissue from the plant, and you must be doing some sort of sample preparation. And if you do some sample preparation during the process, what happens, it will be dried. And then if you expose any sort of technique, or if you employ any sort of technique, then you will be losing some sort of information, because plant is a well-known plant tissues in which a lot of biomolecules, in which a lot of hydrated contents are there, means OH bonds are there. Once OH bonds are got dried, then what happened? Then some sort of molecular bonds are already got damaged, and then by the time you can implement the technique, the whatever the information that you get may be artificial, not in original state. So reason why Raman spectroscopy is one of the promising technologies that straight away you can implement into a native state or a hydrated state of the plant, so that you can extract the chemical composition of any sort of tissue of a plant. That's what the my urge of the plant scientists to make use of Raman spectroscopy in your future research work. And coming to the other issue I mentioned here, to investigate various compositions and structural property, and therefore what do you get? You can derive the molecular information. So when you get the molecular information, and it is very easy for you to implement, and very easy to, of course, in plant taxonomy also, and you classify the plant stuff into different families. And for that, you need to have what sort of chemical composition with which the leaf is made, or the plant tissue is made, or the plant is made, and which one is having some chemical advantage, which is having some pharmaceutical advantage. And you are you will be looking for the plant which is have medicinal important. And if you know the chemical composition of such plant tissue, then only you can exploit and explore of that particular plant into your research career. Otherwise you cannot go for it, right? Otherwise, as a natural science curiosity, you can study chemical composition and you can leave it. Unless you study it in insights, deepen the chemical groups or molecular what species that are present in plant species, then only you can exploit its applicability. And that's what the another issue I would like to emphasize basing on the Raman spectroscopy. And coming to this, and of course, as a physicist, and it's a very new word to use, cellulose crystallinity. As a physicist, I know much about crystallinity, but plant tissues basically having cellulose as a chemical species, right? So therefore, you must know what sort of cellulose crystallinity, and most of the plant tissues are grown in both crystallinity and amorphicity. And you must be aware that X-ray crystallography or X-ray diffraction basic tool that can be implemented to find out the crystallinity size of the cellulose stuff of the plant tissue. So here I'll bring to your information that Raman spectroscopy over any other spectroscopy or any other known as tool to you till today, you can replace it. Instead of using the known technology, you make use of Raman spectroscopy, and you can come definitely with finest cellulose crystallinity by suppressing amorphicity and other kind of stuff. And from literature, I found that Sagel WAXS, WAXS, wide angle X-ray spectroscopy database, US, is one of the standard technique to determine the cellulose crystallinity. And only people I've gone through such a very good reputed journal research papers on plant taxonomy, and I came that cellulose crystallinity that you determine or measure from by using Raman spectroscopy, which is absolutely in agreement with what other devices, what other mechanisms do. So therefore, now onwards, you can also make an alternative to make use of Raman spectroscopy to determine cellulose crystallinity. And not only that, and you have to, as being a plant scientist, you can also quantify lignin amount in a plant cell walls, because plant cell walls are the plant, also part of the plant tissue, and you must quantify it. Okay, so lignin basically having some phenol groups, and phenol groups are also known for its coloring agent for the plant tissue. So therefore, that's what some disadvantage with some sort of spectroscopy called fluorescence spectroscopy. As a plant scientist, you might come across this fluorescence spectroscopy too. So lignin basically, and the basic component is phenol groups, and phenol groups are known for fluorescence, and fluorescence spectroscopy was on alternative techniques to see the morphology of the plant tissue. So here Raman spectroscopy can also be an alternative tool you can look for, and if you really wanted to exploit its technique advantage. And besides it, cellulose water interactions. So this is another advantage of Raman spectroscopy. So cellular water interactions. Why cellular water interactions are required? Because cellulose based stuff can be very widely useful in paper industry, and which of the plant can be very much useful for good quality of the paper, and you require some useful spectroscopy to to study in detail about cellulose-water interactions, and that's where Raman spectroscopy can be dealing with. And not only that, and 2D, 3D images of plant tissues. It's really recent. A couple of years ago only, people started mapping their plant tissues by using Raman spectroscopy. So that's where you can also start imaging the plant tissues by using Raman spectroscopy. And besides, I know I can keep all these in one side. And the other very important thing called the SERS. Of course, later I'll come to that stuff. Surface enhanced Raman scattering spectroscopy is also useful to find out food quality. Food quality in the sense these days is one of the biggest business of the country, biggest business of the world, to adulterate the food. And that's what thereby our health can be deteriorated and our lifespan is also, coming to 60, 65. Earlier, human beings used to live up to 90 years comfortably without having any disease. And since it is food adulteration, and it is one of the biggest challenges for the human being to sustain with a good health. And that's where Raman spectroscopy is one of the promising tools. And it is essence of the Raman spectroscopy is that you can simply, by using handy and a prototype sort of Raman device, within 30, 40,000, you can buy it, and you can just check. Of course, I'll deal with that topic in the end of the lecture. The food adulteration, vegetables and fruits. Not only that, any grain stuff you can take, and you can just expose to the Raman signal, and you can quantify whether the food is original in content or is it adulterated. Okay. And with this, I hope I reach why Raman spectroscopy in plant science is very important nowadays, and why you have to study, why you have to know much about Raman spectroscopy. And besides it, and being Indian, it is also our prime duty, because I always take pride in take up Raman research in India, because it's only the technique we must be proud of, because only the science Nobel laureate from our country and received Nobel Prize in science history as far as Indian scientist point of view. Science, if you take physics, chemistry, life sciences, he's the only one iconic person as far as the science is concerned. Now I'll go with another slide. Of course, already I spoke much about the first slide. I'm not going much detail about the other slides. Just I'll give you glimpses of Raman life and what exactly led Raman to discover Raman effect. One has to have some driving force in his life and to come up with some scientific endeavors, scientific discoveries. And really, what Raman was, what sort of circumstances thrived the Raman to discover this effect. And basically, I would like to highlight much on the fundamentals, because without fundamentals, and most of the other day, the organizers told me most of you are research scientists. Means, of course, besides teachers, there are young research scholars and master students. And as a life scientist or as a life science student, you may be not knowing about the fundamentals of Raman effect, and that's why I would like to throw much light on it. And I'll come to the different types of Raman spectroscopy, and then Raman spectra of few molecules I'll talk about, and the applications, few applications of Raman spectra in plant science I'll come. Of course, and very few important points I'd like to highlight in this. Raman, who is an Indian pride, already told in the beginning, and he was a fellow of Royal Society in 1924. And then if any participant from Calcutta, any participant from Andhra Pradesh, any participant from Tamil Nadu, you must be proud of this great gentleman who made Indian science proud in the history of the science. And then he also worked at the Indian Association for Cultivation of Science. That's where he laid his foundation and led finally to the Raman effect in 1928. And immediately see, once any scientists who do a lot of contributions to the science, it takes many years to get a Nobel Prize. since the Raman effect and its implications in the science and its promising applications in the science, within no time, within 2 years of time, he got Nobel Prize. And that tells the essence of the effect. And later on, Bharat Ratna, 1954. And here I would like to mention, he was the first Indian director, Indian science director for Indian Institute of Science. And of course, it's a very pride for any Indian scientist to become director for Indian Institute of Science in those days, because since inception of Indian Institute, because it is known as Tata Institute of Science, because Tata founded this particular organization with the help of Swami Vivekananda. So therefore, for that, there was a customary that only Tata institutions where the foreigners used to be the directors. And because of Raman, he was a recipient of Nobel Prize, that forced the Tata Group to make him director for Indian Association of Science, and that is also another prestigious positions he held. And later on, once he discharged the duties as director, when he retired as a director from IAC, he established a very prominent frontiers in the science and technology. The great institution called RRI, since its functioning in 48, he established institution, and since till he lost breath, he continued his scientific endeavors there. And of course, you are a young student, and you can also try to have your research career at the RRI, and definitely that can create some sort of scientific, elegance into your career and make you fulfill your dreams to come. And of course, I'm skipping the slide, and I was fortunate that I worked an institution which was in front of the ISSR, and of course, I visited Raman laboratory where Raman invented Raman effect. Yeah, it is puzzle for any life science student. What is a wave? As a physicist, we always deal with waves without wave, and probably it is not possible to deliver any talk in physical sciences or even chemical sciences. As a plant scientist, and you may come across the word called wave, and you may not. So what do you mean by wave? Of course, in order to make you realize, I have shown some reptile picture here also. Okay, when you just sort of throw a stone onto the water surface, what happened? Ripples will be formed. So it was earlier, it was a pea surface, and the disturbance, once you create, the energy that you are creating the water surface can go into the wave. Okay, so of course you have seen these pictures that will make you realize what exactly the wave is. And this is our very important fundamentals before you realize the importance of, before you can understand the Raman spectroscopy or Raman scattering technique. So here I showed you, in the first, I have numbered the pictures because there are many pictures I've kept. The first picture here, the solar system or sun rather, emanating some sort of radiations. Okay, this is called electromagnetic radiation, and there is, I have also shown Earth also, Earth receiving abundant solar radiation every day. And here I have also shown in the picture too. And here we have to deal with electromagnetic radiation. Electromagnetic radiation, you must understand. And if you can understand electromagnetic radiation, then it is very easy for you to understand the consequences of the Raman spectroscopy. So what do you mean by electromagnetic radiation? It's a simple wave, and which is composed of both electric and magnetic fields. Friends, remember, since it is electric and magnetic fields, and it's only electric field component of your wave will deal with the Raman scattering or will deal with the Raman spectroscopy. So now onward, you try to memorize and keep it in your brain that or mind that electric field vector of electromagnetic signal do play a key role in giving you Raman spectra of any sort of molecule. So what do you mean by complete wave? Look at the picture number 3. This is complete wave. From where to where I have to call as a wave, because I have to deal with some properties of wave. Then you must be ensure that what exactly wave is. So therefore, now look at the bottom of the slide. There I've kept some properties. Without knowing the properties of wave, it is not possible for me to deliver entire lecture. So being a wave, it has some peculiar properties. It has a wavelength. I'll show what is exactly wavelength. I'm treating you as a learner and I'm teaching all these things. If you are already well versed with this technology, I know, you please bear with me. If you are a newcomer, try to acquaint with these words. And if it's a wavelength, okay, and the other characteristic property of wave is frequency. The other is energy. And if you know about frequency, if you know about the wavelength, then it's very easy. Of course, I'll also come across with some very fundamental and great principle existing in the physics is Planck's energy rule, that I'll show you how important these three parameters for a Raman spectroscopy is. And now look at the number figure 4, you mentioned 4 here, wavelength. So what do you mean by wavelength? Now look at here, I have shown a wave here, and I have taken a distance, from one crest, means one peak to the another peak, and the distance between them, two successive peaks, is called wavelength. This is very important, what do you say, parameter you must remember and during entire talk. And coming to the number 5th diagram, you look at the red boxes that I mentioned here. In the same space, here I mentioned some low frequency and high frequency. So low frequency, now see, which is having low minimum number of waves, and high frequency, and some sort of disturbance is coming.
1 second, sir. 1 second.
Yeah, thank you. So the red box which I mentioned here to make you classify what is meant by low frequency and what is meant by high frequency. Now see the bottom picture number 5. Low frequency means having low number of, very less number of waves. See that only 2 or 3 waves are there. That's why low frequency within the given space. And if you can accommodate more number of waves, and that's what the frequency increase. So that's what the decrease in frequency, low frequency, high frequency. These two terms are play predominant role in understanding Raman spectra. That's what I'm emphasizing much on it. And now look at picture number 6 here. When I send the white light, and of course you are much aware about any light which is coming from your normal tube light or solar radiation that you are receiving out. That's what when you know these days what's happening, rain is parallelly happening during Mercury day or sunny day. What's happening, beautiful rainbow is seen. How come? So this is what I have shown in the picture number 6. When you send a white light which is composed of 7 colors, starting from visible, means it's also called visible radiation, starting from violet and with the red color. That's what, if I allow the white light to fall on any prism, to fall on any molecule, raindrop rather, and it is splitting into 7 colors. That's what I have here specifically mentioned what you mean by low frequency and what do you mean by high frequency. So here, see here in the bottom of violet color, I have shown many number of waves in the given space. If you are moving from bottom to the top, where I'm ending means coming from violet to red color, with the same space, what is happening? Less number of waves are accommodating in the same space. That's what the meaning is. When I'm moving from violet color to red color, my frequency gradually decreasing and my wavelength gradually increasing. And this is going to play a predominant role in understanding the Raman spectra. Now I'm going to the next slide. And of course, as a physicist, it is very vital to understand the electromagnetic spectra. Of course, if you are a beginner of Raman spectroscopy, if you are well aware with the Raman spectroscopy, it is also a very important slide where you have to throw some light on it. If you move from right hand side to left hand side, you can see the rays I have mentioned in a boxes: gamma ray, X-ray. Already as a life scientist or as a life science student, you have come across the greatest applications and advantages or implications of these rays. X-rays are simply useful to get the bone tissue, the X-ray of a bone where some fractures are lying in the body. That's very useful, and that's what gamma ray. It's also any tumors, unknown tumors are there in a body, and any tissue in a body, any cell in a body. If you can expose those cells to that X-ray or gamma rays, these cancer cells can be destroyed, and therefore your wealth can be restored. You know that. And of course, coming to visible radiation, of course, with only the radiation we can see with our naked eye. And coming to the left hand side portion where I mentioned microwave, FM and AM. Every day we are likely to listen to FM or AM sort of radio signals. That's what we enjoy our music. That's what our cell phone technology and communication technology relying on this sort of electromagnetic radiations. Of course, what is the wavelength and the sizes I mentioned by comparing the size with the atomic nucleus, comparing the atom, comparing the virus, comparing the bacteria. As I'm moving from the right hand side to the left hand side, what happened to the size of the wavelength of a wave? It's moving from atomic size to a mountain size. That's what the beauty of electromagnetic radiations do have different roles to play in different applications. I'm not touching it. And now you look at the very simple equation, which is acknowledged as an equation of science after E is equal to mc square. Specifically, in a physics point of view is concerned, E is equal to h nu is called hc by lambda, playing predominant role in understanding entire quantum mechanics. Hope you heard this about quantum physics or quantum mechanics somewhere in your BSc, somewhere in your intermediate. So relevant stuff also mentioned. And from this one equation, which is equation makes you very clear, nu proportional to 1 over lambda. Mean to tell you is the radiation which is have high frequency definitely have low wavelength. Radiation which is have high wavelength supposed to have low frequency. This is what the relation existing between frequency and wavelength. You must memorize it, right, till the end of the lecture. Frequency is larger, wavelength is lower, and the frequency is lower, the corresponding wavelength is higher. And that's what led Raman to discover Raman effect. Of course, I'm not touching on it since it's already time is running very fastly. And of course, why sky is blue? That's what the Raman tried to figure it out, the Raman effect. Of course, very beautiful understanding by the Raman. Whenever sun sending some sort of electromagnetic radiation, of course, as a lifetime scientist, you are aware that if you move from the Earth's surface to 500 kilometers, and where your iono, strato, tropo layers or spheres will be end up, then there is no radiation. That's what it looks black, though we are receiving radiation. So that's what electromagnetation not require any medium to pass through the space. Once they enter the atmosphere, what is happening? Scattering takes place. Scattering is a very important what to realize. When the electromagnetic, that's what see in the first picture I have made you realize. First picture. In picture number 1, you're aware I've put my cursor. When solar radiation entering it, and of course you know that environmental scientist as a botanist is also equally known as an environmental student or environmental scientist. You know that environment which is composed of many number of molecules: nitrogen, oxygen, hydrogen, helium, lower fractions of argon, and some sort of stuff. So therefore, the moment electromagnetic radiation enter the Earth atmosphere, then what is happening? I said electric field vector of electromagnetic ray try to interact with the molecules. When the electromagnetic radiation interact with the molecule, what is happening? When somebody is impeding your motion, what is happening? When you're running, somebody is trying to impede your motion, then what's happening? You just take the left or right turn and you move, right? So that's what the scattering does. So similarly, electromagnetic ray, light travels in straight lines. The moment it enters the atmosphere, where it sees any molecule interacting with electromagnetic signal of the EM radiation, that's what it takes its path from straight line to another path. That's what see in the picture number 2 I have showed, sorry, picture number 3. When red color going straight away means it is not deviating. Yellow color is also going straight away, and the green color starts going straight away. Mean to tell you, these electromagnetic radiations are not playing or not interacting with these molecules, because the energies the molecules needed is not the red one, not the yellow one, not the green one. So therefore, the energy the molecules needed is blue or needed is violet. So therefore, since violet and blue color having relevant and high energies, so therefore the molecules try to interact with that. So once they interact, what is happening? They get scattered. That's what the entire blue color, entire violet color can be spread out throughout the space. So since violet is occupying very narrow space on EM spectra, you cannot, because since a long distance the violet color travel and your naked eye cannot see that. So ultimately we are gonna see and realize the entire sky looks like a blue. Then this is what happens when during the interaction or scattering of blue color light with the molecules of environment. This is what scattering means. And of course, fourth diagram is also very clear for you, and rest of the radiations are unaffected, and only blue color light get affected and spread across the space. That's what you and I realized, that the entire space looks like a blue. And of course, I'm not going in detail about it. And where it's a very interesting story that I like to emphasize. J.C. Bose and one of the plant scientists. And what is the link between J.C. Bose and Rayleigh? Rayleigh is a great scientist and physics and chemistry point of view. And Rayleigh is one of the backbone in discovering argon element. And of course, you realize what is importance of a life scientist. And if you're doing some of the inert environment experiments, and you can realize the importance of argon gas. And he was inventor of the argon gas. And J.C. Bose did his PhD under Rayleigh. That's what they, J.C. Bose did fantastic contribution in radiation science, and basically he was also inventor of radio, right? Of course, Marconi later on, he was named himself, and J.C. Bose one of very important personalities. Of course, he didn't patent it, he didn't publish any of his data on radio science, and that's what he didn't get the required recognition in the science, in especially physics. And of course, he got equal reputation in plant science, and you know much about J.C. Bose than me. And here, these gentlemen, Rayleigh and Raman, okay, contributed nicely towards the scattering, and I'll make you realize what important contribution Rayleigh and Raman did towards the scattering. And I'm not reading it right now. And now, look at here. Here first I'm not taking any complex molecule right now, because if I straight away take you to the complex molecule, makes you a technique which is more ambiguous. So therefore I took here only diatomic molecule. Hope you know about diatomic molecule. Diatomic molecule, nothing but only two atoms, maybe of same kind or two different families, right? That's what you can call homonuclear and endonuclear molecules. So therefore, if you take a diatomic molecule, which is called heteronuclear here, because I've taken silver globe and red color ball here. Then what is happening when these two atoms are tied together? There is a chemical bond. Then what automatically they'll come closer and moving apart. So therefore, they start vibrating each other. When they start vibrating, what is happening? And individual atom having energy levels, and besides it, when they form these two atoms come closer to each other, and therefore they form a molecule. When they form a molecule, what is happening? And besides the electronic energies, they also possess the vibrational energies. So vibrational energies do play a very important role in order to characterize the biomolecules. So that's what the picture is very relevant as far as the biomolecule point of view. And that's what I've shown the Morse diagram also, how a molecule can possess different vibrational energies. I have shown here V is equal to 0, 1, 2, 3, 4. As soon as the two molecules are moving slowly, then they have lower vibrational energies. Moving very rapidly and then fastly, their bond lengths are increasing, and automatically their bond energies are increasing, their vibrational energies are also increasing. In that stuff, I have included this slide. And here, this is another diagram which I want to emphasize. So see here, Raman effect, which is very clear here for you. In the left-hand diagram, electromagnetic radiation, which is of some frequency. And if you take some visible radiation, like, of course, green color radiation I have shown. So green color having 5,500 angstrom, because angstrom is a unit to measure the wavelength. Already I've made you realize what exactly wavelength is. So therefore, it is having its equal frequency. It is having some 5,500 angstrom unit wavelength. And with this wavelength, if it is falling on a molecule, this is the molecule here I have shown, diatomic molecule, right? This is diatomic molecule, 2 atoms, and which if it is exposed to this radiation, then what is happening? The scattered light, because light which is falling on a molecule, definitely if it is molecule is capable to absorb, it's okay, the light cannot come out. But it's not the case. The molecule cannot absorb entire radiation. It can absorb only few amount of radiation. The rest will come out as a scattered photon. And therefore, the scattered photon comprising of 3 important radiations. How many? 3 important radiations. One is as it is. Suppose if I allow 10 number of what do you say, green photons. So because photon is a relevant word to be used, it's a one wave. So how many number of waves I am allowing to fall on a diatomic molecule? Let us assume 10 I am allowing. So out of each 10, how many? Maybe there are 7 or 8 photons come after interacting with the molecule, come as it is. Means with the same green color, it come as it is. That's what it is called Rayleigh scattering, unmodified radiation. So what do you mean unmodified radiation? Though it is interacting with the molecule, nothing it can lose. Same, whatever the energy, whatever the frequency, whatever the wavelength it is impinging on, it comes out with the same sort of wavelength. That's what it is called Rayleigh scattering. Okay, I'll go back to the previous slide. Now you see that's what I have written. You can also read Rayleigh scattering. So the incident radiation and scattered radiation, incident wave and scattered wave will be same energy. That's what it is called Rayleigh line, Rayleigh scattering. And coming to this, Stokes-Raman scattering and Raman scattering. Now I am in a position to define Raman. So Raman is when you allow electromagnetic radiation to fall on any sort of molecule. Molecule may be biomolecule, molecule may be inorganic, molecule may be organic, molecule may be in gaseous state, liquid state, plasma state, solid state, whatever the state, irrespective of the state. If there is a molecule, there is a Raman spectroscopy or Raman scattering. Then if this particular photon loses some amount of energy to the molecule and scatter with a lesser frequency, then this is called Stokes-Raman scattering. Suppose the incident radiation interacting with the molecule and scattering with high energy, means during interaction, the radiation takes some amount, because every molecule having energy, and therefore during interaction what is happening? Instead of losing the energy to the substance, instead the incident radiation capture energy from the molecule and scatters. That's what it is, high energetic beam. So therefore the modification during the interaction, if there is a modification in the energy of the incident radiation, is called Raman scattering, and this effect is called Raman effect. Once again, I'm defining the Raman effect is defined as when you allow the electromagnetic radiation to fall on any sort of substance, if the incident radiation either gaining the energy and scatter, either losing the energy and scatter, and that's what the phenomena is called Raman effect, and that's what the scattering causes, Raman scattering. And why? Because it's every time my mind is blowing that how come the scientists, because we have seen so familiar, so popular scientists up to 1928. You also know the number of live scientists who have done path-breaking research, okay, and groundbreaking research. Why that phenomena left to the Indian scientists till 1928? Here is the beauty. Here I have mentioned some bottom line in a red color, okay, brick red color. You read it. So because of this particular reason, and scientists till 1928, nobody recognized the effects. So there is, if you take 1 million photons to 10 million photons, 1 million means 10 lakhs photon. If you allow 10 million waves, 10 million means electromagnetic signals, means are 10 lakhs or 1 crore electromagnetic waves to fall on the molecule, then only how many Raman photons will be observed? Only 1 Raman photon will be observed. Then is it a strong effect or is it a weak effect? It is a it's a very, very weak effect. It is vulnerable effect rather. That's what this phenomena waited till a person who can do a lot of striving to get that particular effect. And this was only possible with Raman because he did lot of experimentation from 1922 to 1928. Altogether 6 years he carried experimentation. That's what he paid the price with the effect called Raman. Okay. And later on, 1924, he was called by Rutherford and J.J. Thomson. Probably you heard about these two gentlemen. One was the inventor of the photon, Rutherford. One was the discoverer of the electron, which are the two important constituents of an atom. These two great gentlemen hold a science congress in UK, London, and they invited this Raman because of his experiments in 1924. And he then went to London and he deliberate some sort of interactions with the molecules and scattering. Of course, by the time Raman effect was not, what do you say, was not invented. So therefore, they encouraged him a lot, and then finally he came up with this effect. That was his great beauty. Now you see, you will be wondering by look at this diagram number 2 here. So if you have a molecule and if you excite some sort of radiation, then what is happening? If you excite a molecule, where does it go? The electron will go from ground state to the excited level. But in the Raman scattering, it is not. When you excite a molecule, instead of going electrons, a molecule, instead of going from ground state to excited level, it will go to intermediate level. That's what is called virtual energy level. So of course, I'm not talking much in details about it. You remember virtual level. Virtual level is only created when electromagnetic radiation interacting with the molecular stuff, and therefore the molecule will come down and will get scattered away and will give you a Raman effect. Now see a quantum mechanical treatment. I'll focus 1 minute on this. Rayleigh scattering I already talked about. Okay, whatever the frequency with which it is incident on the molecule, the same energy, same wavelength, it scatters out. But in the case of Raman scattering, only 1 photon out of 10 million photons, it sees some sort of change. It goes from one state to the see the second line diagram here, where I've kept the cursor. It goes from lower vibrational state to the virtual state. And in the returning, what is happening, red color I've showed. Okay, instead of coming to the E0 level, it end up with E0 plus H nu level. And that's what the some sort of energy it's already given to the molecule, and the rest energy it scatters. That's what here in the second diagram, where I've kept my cursor telling you a big green color is Rayleigh, the left side red color showing Stokes Raman, which is having lesser in frequency. The blue color is showing a high frequency line called anti-Stokes line. That's what, when you're talking about Raman spectroscopy, you must talk about both Stokes and anti-Stokes line. And coming to this, I know Raman, though he was working at ISSR and he did not have money, that's what he requested J.R.D. Tata to fund him. Okay, so J.R.D. Tata funded him 600 rupees, out of which he bought the spectroscopy, which I have shown here from London, and he carried out experiment. In written, he wrote a letter to J.R.D. Tata and said that for 600 rupees he brought Nobel Prize. And those days 600 is worth of bringing Nobel Prize to the country. That's what very committed researcher Raman was in those days. See a beautiful spectrograph that Raman used, and how the scientists surrounded Raman to see the Raman effect in those days. And of course, it's not an issue. You cannot manufacture Raman spectrograph, Raman spectrometer in 600 rupees. And it has gone up to 3 crore rupees right now. And I'm pretty sure, and most of the Indian institutions are not even in a position to buy Raman spectrophotometer, because that has brought in the technology by the foreigners, completely grabbed the Raman effect and started manufacturing. Very big champions in the industry of Raman machines, and they are developing. And most of the IITs right now catering the needs of Raman spectroscopy, and ordinary institutions not in a position to buy it because it has gone up to the 3 crores rupees. Why I have told you, there is only one Raman photon you have out of 1 crore, electromagnetic radiation that you are allowed to fall on. So therefore, rest of the photons you have to eliminate, you have to filter out, then you have to identify that particular one photon. It is very tedious process for any technologist. That's what the price has gone to 3 crores. Okay, now that's what the very state-of-art machine I have represented, I have shown in the slide. Of course, very salient features. I'm not going in detail right now. I'm just skipping the slide. Yeah, in 1928, and I'm very sure not even Raman, anticipated that his effect gonna break the fresh grounds in the research or mind-blowing research the people can come up, can be used widely in all life sciences or physical sciences, material sciences, because he just anticipated any physicist can make use of to study interactions of the molecule. With that, envisage, he invented effect 1928 with a white light. And now with the invention of laser by Townes and Maimon, both are US scientists, 1960, with the invention of the Raman lasers, this has made Raman effect as a universal effect and has become powerful and powerful Raman scattering, and it has been reaping out the many fruits and many industries and many applications in the sciences, and especially in a crime investigations, Raman spectroscopy playing a fabulous role. Okay, you can realize in the end of the slide. And now, for any youngster who is unknown about how to plot, and suppose if you can Google it, you can, it very easily shown a Raman spectra of any molecule. Peculiar molecule is shown x-axis Raman shift, and the y-axis it is intensity. Okay, and therefore Raman shift is values should be taken on x-axis, and Raman intensity corresponding photons. How many photons are detected by your CCD camera of a Raman machine can be taken on a y-axis. So this is basically, if you have data x-axis and y-axis, you can plot in Origin software. Of course, Origin software is freely available in Google. You can just Google it. You can have a download free version, and you can start just plot the Raman spectra of any molecule of your concern. So what do I have to emphasize? A very important word here, Raman shift. What do you mean by Raman shift? Here, right hand side diagram I've shown here on Stokes Raman. See here, sorry. See here, in the case of blue and green colors, in this basically whatever we measure is a Stokes Raman scattering, anti-Stokes Raman scattering, which is very, very weak effect, and you cannot measure it unless you have a very specified Raman spectrography or spectrometer. You cannot identify, because of the way we are using spontaneous Raman scattering, where we can people developed it only based on Stokes Raman scattering. So therefore, blue color you see, green color you see, there is a some difference here. I have mentioned in a Raman shift is the energy difference between incident and the scattered light. And if you can, of course, machine can do that, but you have to understand what exactly Raman shift is. Raman shift is you just take the energy of scattered photon and you take the energy of incident photon, and you just subtract one energy from the other, and you get a qualitative amount, and you get a magnitude of it, and that is nothing but Raman shift. And those values you take on the x-axis, and preferably that energy is measured in terms centimeter inverse. You will be wondering, because you understood energy unit in intermediate or UG level, that electron volt or joule or erg. Okay, still centimeter inverse, which is a very, very tiny, what do you say, unit people are using. That's what joule is a very big unit, erg is a very big unit, electron volt is a very big unit, because molecules are having very low energies. That's what centimeter inverse has become important dimensions for a Raman scattering scientist to measure the energy of molecule in terms of centimeter inverse. That's what centimeter inverse has come into picture. And now see homonuclear, heteronuclear molecule. For your reference, I have shown what do you say, heteronuclear, homonuclear molecules. I have started with a simple molecule and I've ended with a polyaromic molecule. See here, H2, O2, N2, very known as a homonuclear. Homonuclear means same sort of family atoms. They just covalently bonded, and they are making you diatomic, triatomic. Like if you take O3, it's a triatomic molecule of same family. It's a homonuclear molecule. But if you take the right hand side picture, what I have shown here, polyatomic H2O, if you take, it's a what do you say, heteronuclear molecules. Though it is a triatomic molecule, two of the oxygen atoms, one hydrogen atoms combined together, and they are making a triatomic molecule, which is called the heteronuclear molecule. Why I have to learn about homonuclear and heteronuclear molecules, you will be realizing in the next slide. Now you see, in most of your plant tissues and most of your plant chemistry, if your chemical compositions of your plant tissues are concerned, and they are basically composed of alkaloids, and alkaloids are basically function of carbon and nitrogen hydrogen bonds. And of course also plant tissues are also having cellulose fibers, and cellulose fibers, and plant cells also cell walls specifically concern lignin. Already said phenol groups, of course phenol group is also shown in the picture. So therefore, what you commonly understood that in every functional group of your plant tissue is either composed of carbon groups, either composed of hydroxyl group, OH groups. That's what it is. I feel relevant to talk about Raman scattering for H2O molecule and CO2 molecule, and later I wanted to extend it to the further molecules. Now you see a beautiful H2O molecule here I mentioned, because unless you understand this, you cannot appreciate the Raman scattering. Okay, so therefore H2O molecule here I have shown in the top right hand side, top left hand side picture where I have kept my cursor. H2O, because hydrogen having one electron, one proton, hydrogen also other side hydrogen, one electron, and it is sharing covalently bonded with the oxygen. And of course, for your information, oxygen having how many atomic number, or what is atomic number of oxygen? Oxygen atomic number is 8, and of course 1s2 is completely bonded, and 6 electrons are left for the bond. And of course only to get an octet state, of course any molecule to get a stability, it goes for octet state, and therefore the electrons present in the oxygen donates its electron, one electron to the hydrogen and another electron to hydrogen, and therefore it is covalently bond. So therefore what is happening, there is a sharing of the electron charge from hydrogen and with the oxygen. During the sharing, what is happening, the charges are transferring from hydrogen to oxygen and oxygen to hydrogen, and therefore what is happening, they are no more neutral. Of course you heard about neutral charge. Almost every atom, every molecule is neutral, right? Because there is no charge excessive. But if you focus on individual atoms during the formation of the covalent bond, ionic bond, any sort of metallic bond, and they'll undergo some sort of change in the charges. That's what I put in diagram number 2 here in a purple color where I kept my cursor. So therefore here I have written that delta plus delta minus. So once the electron lost by the hydrogen to the oxygen, that's automatically what is happening, it is becoming positively charged because it lost electron to the oxygen. And since electron acquiring the 2 electron from 2 adjacent hydrogens, it acquiring the negative charge. So therefore what is happening, a negative charge is accumulated on the oxygen and positive charge accumulated on the hydrogen. Now we come to the third picture I have shown on the right hand side, okay, right corner. That's what when two charges, positive and negative, separated by some distance, what is happening according to the physics, the molecule or the atoms gain a property called dipole. Now the atom behaves or molecule behaves as a tiny dipole. So therefore the tiny dipole having a property called dipole moment, right? And of course if you appreciate me if I tell this, and most of you are also making use of IR spectroscopy. IR spectroscopy is a very important tool to study the vibrational structure or vibrational spectra of your plant tissues, right? So therefore, for any molecule to be IR active, what is that? The dipole moment, net dipole moment of the molecule supposed to be not zero, right? So that's what the dipole moment comes out here. Similarly, in similar lines, a molecule to be Raman active, the molecule supposed to have, though the molecule need not to have dipole moment, net dipole moment I'm talking about. So it's a dipole moment. Though the molecule do not have net dipole moment, that molecule you can characterize, that molecule you can study by using the Raman spectroscopy. Why? Then I'll come to the next slide. So therefore, of course, CO2 molecules I presented here. Now come to this. Some sort of pictures also I kept, how charge separation can be taken place between hydrogen and oxygen and carbon dioxide also I've represented here in the bottom picture on the right hand side corner. So carbon sharing its 2 electrons with oxygen, so therefore carbon becoming positively charged, oxygen becoming negatively charged, therefore dipole moment is present in this. Now I'll come to the next picture, you will be realized. Yeah, now here electromagnetic signal already I told you, right, in the beginning of my talk. So when electromagnetic radiation is falling on H2O molecule, what is happening? Electromagnetic signal having positive and negative, right? Because it is electric field. Electric field is nothing but having positive and negative, right? If it is going in an upward direction, it is a positive cycle. We have to nomenclature, we have to call it as a positive cycle. If it is going downward side, it is called negative cycle. When the electromagnetic signal simply passing and interacting with the molecule. See in the this picture here I have shown in the second diagram number 2, which is in the left hand side where I've kept my cursor. So during the interaction of the electromagnetic signal, depending upon the means negative cycle, positive half cycle, what is happening? The places of the hydrogens and the places of the oxygen changes accordingly, right? So therefore, their dipole moments will be changing according to the electromagnetic signal, the way it is interacting. So therefore, what is happening? The bond, because of electromagnetic interaction, the bond length of your hydrogen oxygen will change. So when it is changing, automatically it changes the bond length. When bond length changes, automatically the induced dipole moment comes out. And you see in the third diagram now, the right hand side portion I have kept here my cursor. Okay, my cursor. So therefore, when it is interacting with a substance as a whole, then dipoles are aligned along the field direction, okay, according to the direction of the electromagnetic signal. And finally, there is a property called polarizability. Polarizability is nothing but the dipole moment is already represented by mu proportional to E. Electric field changes, electromagnetic signal varies, and automatically dipole moment changes. When dipole moment changes, the polarization changes. In turn, the polarizability of the molecule also changes. Therefore, the polarizability in the picture number 4, right hand side, the bottom picture shows where I kept my cursor, telling you alpha is an indication of polarizability, the dipole moment per unit electric field. When you are changing electromagnetic radiation, accordingly dipole moment changes, thereby there is a structural change in the dipole, and therefore any molecule which is having polarizability, and the molecule must be Raman active. Now see here I mentioned a mathematical concept, d alpha by dq not equal to 0. What is alpha? Alpha is polarizability with respect to q is the distance between two molecules. If that is not 0, then that particular molecule is Raman active. Yeah, see here how N2 molecule and O2 molecule. Since here I would like to mention very important point, H2 molecule, O2 molecule, you cannot characterize by using IR spectroscopy. Why? H2 molecule having no dipole moment, no net dipole moment. Though they are dipole, they are having the heteronuclear molecule. So the heteromolecule molecule you cannot characterize because net dipole moment is always 0, and therefore that molecule you can characterize by using the Raman scattering. That's what here I represented. Now say CO2 molecule. CO2 molecule is having 4 sort of vibrations. Okay, because of course you are not a physicist or chemistry to realize the normal modes or whether it is linear molecule, non-linear molecule. Okay, H2O is a non-linear molecule. Depending upon that, it has 3 type of vibrations. Okay, and stretching, symmetric stretching, anti-symmetric. And the CO2 molecule, it is a linear molecule, therefore it has 4 type of vibrations, 2 bending vibrations, 1 is symmetric stretching, and other is asymmetric stretching. What do you mean by symmetric? Symmetric means centers, carbon is not moving, oxygens are moving, coming towards carbon and going away. Asymmetric means one oxygen coming to carbon, the other oxygen going away. This is asymmetric stretching vibration. Of course, here I mentioned in the diagram also. I'm just quickly going because time is already whatever time has given to me, maybe 4:10 or 4:15. I'm already surpassing the time. The see here, you can appreciate Raman because Raman only invented spontaneous Raman spectroscopy. Later on, people developed so many spectroscopies, starting from resonance Raman, stimulated Raman, coherent anti-scars, playing very important role in your plant science. Please try to learn about it. And surface sensor Raman scattering. Of course, 1 or 2 minutes I'll spend my time on surface enhanced Raman spectroscopy, which is playing very important role. Tip enhanced Raman spectroscopy. Individual spectroscopies having individual machinery. Remember, okay, how the science and technology or manufacturers are making money out of our indigenous technique called Raman technique. Of course, Indian companies are not manufacturing any sort of devices because a lot of electronics should be associated in it. Therefore, none of the Indian manufacturers are manufacturing this machinery. Now come to this very important point I have to address here. And since the lasers, visible lasers, you come across many lasers, right? So far, you know what exactly visible lasers. You have seen red color laser, blue color laser, all visible lasers you have seen. But if you use visible laser to study molecular spaces of your plant tissue, it doesn't work. Why? Why? How come it cannot work? Because fluorescence playing a very dominant role. What do you mean by fluorescence? Okay, so here I mentioned the fluorescence. What do you mean fluorescence? If you excite any sort of tissue, what is happening? The electrons in your plant tissue will, or molecules in your plant tissue will excite it from ground state to the excited state, and after taking some sort of nanoseconds, the electrons again moved from ground state to excited state, come back to the ground state. While it is coming back, what is happening? Again they'll emit. Once they emit, what is happening? They will be colored. Once they'll come out, what is happening? They are in turn, they're heating your tissues. If they heat it, the plant tissue will get damaged because they are so soft, right? So therefore you cannot use any sort of laser and to study plant tissues, because fluorescence is the biggest threat for the plant science or plant tissues. So therefore what you have to use, you have to use the neodymium ag laser, which is IR laser. Of course, as a physicist, I can make use of Raman spectroscopy by using any sort of laser to study my chemical compositions, because my materials are very solid in state or liquid in state, gas in state. They won't destroy the structure of my molecules. But your molecules are thermal effective. They are so sensitive to thermal property. So therefore they can damage your tissues. So therefore instead of using that, what you have to use? You have to use IR radiation, because IR radiation having low energy, they cannot damage your tissue, though, because IR is known for its thermal imaging. So therefore though it creates some sort of thermal effect, by using some other technology, you can filter out those thermal effects, and you can safely work out by reducing the wattage of the laser ray, and you can comfortably work and get the data whatever you require from plant tissue. That's what 1064 nanometer, which is FT Fourier transform Raman spectroscopy, and which is a neodymium ag laser, if possible. Okay, if you are working any NITs, and of course most of NITs having this sort of laser, this sort of IR spectroscopy, you can go and visit and you can work with that. Yeah, now I've come to uses of Raman spectroscopy, how you can implement. Here I've shown a plant, and then what is a Raman spectra, a typical. And then I've taken some Raman spectra from one article which is recently published by Agarwal group, which may be UK group. And what they have determined is they have determined cellulose crystallinity. So here you see here top portion, sorry, the right hand side where my cursor is showing here, they have identified very intense peaks, 380 centimeter inverse band and 1096 band, 600 centimeter inverse band, and they have found out some equation where they have estimated the intensities. Of course, origin software I told you in the beginning. So therefore, in a couple of slides after I've showed what is the origin software. So therefore, by using origin software, there are some different fittings. You can fit the peak, and they can extract the peak intensity. So peak intensity treated as area, and the peak intensity areas you can calculate, and you can fit in this equation where I've shown in the C Raman and cellulose crystallinity in the bottom. So by using that, you can estimate cellulose crystallinity, and beautiful, reputed papers, reputed journals are publishing this kind of data. And if you are a young researcher, please keep focus on this issue and try to come out finding cellulose crystallinities. And don't use X-ray crystallography. You can instead of using that, X-rays are so high energetic and they can destroy the tissue, and therefore making use of Raman spectroscopy, you can alternatively determine cellulose crystallinity. And of course, I don't have time to read out, because this is one of the slides which can help you where are the plant tissues and which of the areas, whatever the fingerprint, because I've shown you 50 centimeter inverse to 3000 centimeter inverse, which is a fingerprint zone of the Raman scattering spectroscopy, on which the Raman spectroscopy works. And therefore your different zones, different molecules functions, different molecules vibrates. So therefore this slide will take you which of the functional group present in your molecular species or plant tissues in which you have molecule vibrates. And by looking at this, you can find out whether C double bond C is vibrating, C double bond C is vibrating, NH amine groups are vibrating in your plant tissues. Okay, I'm not going on this. And of course, one of the superior application of the Raman is SCRS. This is called surface enhanced Raman spectroscopy, and which is one of the powerful tool in the nanoscience and nanotechnology right now. And most of the material scientists and what do you say, zoologists and botanists, or even medicines, or even what do you say, crime in the crime branch, neuroscientists can also be using this particular spectroscopy, which is of course I'll show you, which is portable Raman. Okay, this you can buy it and you can come out with this technique. Now you see the superior application, because though there are many techniques, because of lack of time I cannot take you to all this sort of technique, but it's only a useful information which can throw some sort of light. Here in the left hand side portion of my slide, where I kept some apple, right? So what do you do? Suppose if it is apple which is coated with any sort of pesticide and any sort of contamination it is have or adulterated, and in order to estimate the quality of the fruit, then what you have to do is it's already ready-made substrates are available, and it's a glass substrate, simple, and which what is happening, you have a silver coated, or because silver which is a nanoform is coated, or copper which is a nanoform is coated, or platinum or gold especially. Okay, of course silver, copper are cheaper than that of platinum and gold, so they are readily available. What they do, so nano metals will be coated on a glass slide. You just go to the market and buy it, and you just peel the top surface of the any food that you want to test the quality, whether they are adulterated or not, and you just few molecules, you just peel it out from the stuff what you want to testing. Out of it, you paste on some using adhesive tape, you just stick those molecules and you expose it to the prototype device I showed you, right? Therefore you just get a you already know what is a pure apple can have sort of Raman spectra. If it's adulterated, automatically different sort of Raman signal it can exhibit. Depending upon that, you can characterize what sort of chemicals which are applied on it, how much it is adulterated, up to what layer it is adulterated, you can read out. And not only that, right hand side picture I showed you different okay, whether it is organic vegetable or it is no pesticide based contaminated vegetable, you can also read out by simply studying original Raman spectra of that sort of stuff, or what do you say, contaminated based Raman stuff of that particular material. And of course here I have some other food grains also. Most of the food grains, in order to make you attractive, they will be colored, so they will be contaminated, adulterated. In order, whether it is in pure form, adulterated form, you just take the grain, okay, one grain, and expose it to the Raman signal, and we can appreciate it whether it is contaminated or adulterated or not. And a fourth slide, fourth picture, it is also any sort of cool drink or any sort of milk, especially milk. Of course, cool drinks having many contamination pesticides, you don't worry about its quality and purity. Also, milk is one of the prime concerns of human being these days. Most of the news channels are showing that most of the milk industries, milk companies are adulterating it, whether it is a pure, okay, instead of finding out lactose and every other some kind of stuff, you can also estimate what type of adulterated or what type of chemicals are involved into this kind of stuff by simply finding out Raman spectra of that particular milk. So this is portable Raman. It is cheaply available in the market. Just buy it and test for your food safety. Yeah, hope I've come to an end. Thank you very much for your patience listening. Of course, though it is Raman spectroscopy, which is not well known to you as a plant scientist, and I hope I made you realize the importance and fundamentals of Raman spectroscopy on one hand, and what is the important things that the Raman spectroscopy can do in a human life, and besides that in a plant science Raman spectroscopy role. And therefore hope I satisfied in a bit extent, not to the extent that you envisaged, and hope
yeah,
go back.
thank you Upender. It's a wonderful presentation, very informative, highly energetic afternoon session, I believe. definitely it is useful for the plant science, life science teachers and students. We hope they can find a new a way in identifying different things.
It's really a new avenue also for any beginner. For those who are doing in a plant science, it is a really new avenue. They can start off making use of Raman spectroscopy.
Yeah.
Though they have seen Raman spectroscopy only useful for some sort of zoologist or only chemist or physicist can make use of. So now onwards, maybe plant scientists can also make use of it.
Yes, definitely.
I also welcome any sort of criticism or any sort of questions. I welcome.
Please, if anyone is having any queries, please raise your hand.
If there are no questions, means they have not understood anything or they have understood complete.
No, it's a wonderful session, Dr. Upender. Really, you mesmerized our audience because 130 audience stayed with us and they are sending feedback. It's a really wonderful session and
thanks a lot.
See, one of my students is working with the Raman spectroscopy, doing a pollen analysis, and I thought while framing the structure of this course, why don't I keep this for our audience attendees? So immediately I contacted Dr. Upender. He accepted and given a wonderful presentation. Excellent, extraordinary.
Thanks a lot, sir, making me a part of this. And though I'm not aware of plant, and to your surprise, taxonomy is the first time I learned a word. And
exactly.
in the beginning, I really frightened. How can I, exposed to such a learned August gathering, a learned, scientific community. And at the end, at least I convinced you. Thank you. Thanks.
And Dr. Upender also joined as a participant throughout the course and learned many things in botany.
And even using the plant tissue, plant cell, on all this terminology I learned from before speakers. I thanks a lot to the previous speakers also.
Sir, someone raised their hand. I think they want to ask the question.
Yeah.
Hello, sir. Good evening.
Yeah, good evening, gentlemen.
Sir, I have a question. Can we use Raman spectrum in the forest ecosystem, sir?
Yeah, why not? You can use it. Forest ecosystem means where you want to exactly point out the application of Raman spectroscopy.
In the evergreen forest, means interior forest.
No, forest, Raman spectroscopy, nothing to do with the forestry. But any molecule or any species, plant species you want to characterize, you want to study the chemistry or you want to study taxonomy or relevant stuff to that particular plant available for you, or you want to study about because Raman spectroscopy, which is very specifically designed to study the structure and chemical composition of molecular species. And
yes,
if you really want to test any sort of plant, and yeah, definitely you can make use of.
Okay, sir. Okay. Sir, is it available that what you're showing in the?
Yeah, means you're talking about machine or device.
Yes, sir. Machine.
Machine is available in Usman University, in the University of Hyderabad, IIT Hyderabad, and of course I'm talking about Hyderabad, IACT, NGRI, CCMB, and every high standard institutions, not very ordinary, because I told you it is worth of 2 crore, 3 crore. Not many institutions can bear it, but yes, very high institutions where the PG courses and research is going on, those institutions having this stuff, and you can write a mail. And not only that, the UGC having around 8 to 10 centers across the country. Especially University of Hyderabad, there is a nano center established by UGC. It is free of cost. You can just go there directly. You can make use of it. Just put a mail and request them. If free of cost, they can give you Raman spectra of numerous samples and n number of samples. If you have, you can make use of it. You can get the free of cost data.
Okay, sir. Thank you.
If you belong to Hyderabad.
Yeah, madam.
Sir?
Madam.
Hi, yes, sir. Good evening, sir. Upender, sir.
Good evening, madam. Nice to see you.
You gave a very Great lecture, sir. Actually, as you said earlier, that we don't know much of physics. We know very little of physics, especially these Raman spectra and all these things. We know very little, sir. But even to people like us, you made the content, you made the stuff very easy, sir, to understanding and all. And another thing is, you gave a very broad scope for the research, that even we biologists can also have the sampling. Like especially I am from environmental science, so we will be working on these adulterations and all those things. So definitely you have given good information regarding how to use this Raman spectra, and you said about the portable machine, portable Raman I would like to know actually what will be the cost also, sir. Around
50,000, madam. You can get the Raman portable machine. You can get within 50,000.
Sir.
But it is a limited application. It's very limited materials only you can work out. In order to work out, research point of view, because you need a very high-end machine basically. But adulteration thing, that hand machine, portable machine, whatever showed, that is enough, ma'am.
Okay, sir. Okay. For initial research, it is okay. But for the real research, yes, we
in order to explore
have.
it, in order to explore, and you want to exploit much for the research, and you have to go with very high-end machine. It's high-end machine is required. And for your kind information, our CFRD also, you know it, no, ma'am.
Yes, sir.
CFRD is also already tendered, I think, for Raman spectra, and very soon we are going to get very high-end machine. And that will be,
we are very happy and fortunate, sir.
And we can work on it. Basically, we can make use of those CFRD facilities in days to come.
Okay, sir. Thank you very much, sir.
Thanks.
And another, in the last your this thing, you have also said that University of Hyderabad is also having a center where we can have free center. This information is very, very valuable, sir. Everybody will be using that information, sir, because we are in need. Nowadays, the funding is very meager from very few are getting funding. So if we have such free sampling techniques and
I can also give you some sort of information about the Nano Center, which is there in University of Hyderabad by UGC, ma'am. As a faculty, we can go there and we can collaborate with them, with the scientists, life scientists, life science faculties are there. No, just go to them and talk to them. You can also stay for 1 week. As a faculty, we can stay. That provision is there. That privilege we have. Our students can also stay. Suppose some students want to in situ measurements. In situ measurements you can also carry out by staying there for 1 week, 10 days, and they can also give you all the TADA and everything they can give you. That kind of facility we have with the center in University of Hyderabad.
Oh, thank you, sir. Very great information, sir. Really, we don't know this. Definitely, in future, definitely in near future itself, we will be coming to you, sir, to have such collaboration initially with you and then with University of Hyderabad, sir. Okay, thank you very much for having you here, sir, and you enlightened our participant till now. 125 participants are watching you and inspiring with your talk, sir. Very kind of you and very.
And indeed, I'm grateful to all the participants who bared me, though they are not from science, physics, especially.
Yes,
thank you. I must
thank you, sir. Thank you very much, sir. Thank you, sir.
And finally, just let me thank especially Vijay Bhaskar Reddy sir, who always motivates me, and then of course, besides. Last but not least, Raman sir is a very good friend of mine. All the way encourages me a lot. Though he's a scientist from geology point, and so he's a very reputed faculty from botany, reputed faculty from geology. My long association as a physicist goes with them. Hope one day we'll finally we'll do some good science in a multi-dimensional scope. Thank you.
Sir, someone posted one question about session, sir.
Yes, sir. I will come. Welcome. I'll take that question.
Is it possible to identify biological molecules just like AAAs or mass spectrometry?
Definitely. You can unknown if though is unknown molecule is there, because I showed you some slide where the molecules depending upon where that particular molecule showing the characteristic band. With that you can estimate the quantity of that particular unknown molecule. You can estimate, you can quantify it. That's what I am talking about, lignin quantification. So by using Raman spectroscopy, you can quantify how much of that particular sort of chemical functional group is present in your species. That sort of analysis is possible.
Sir, one more question from ma'am, Jyoti ma'am. Jyoti ma'am, you can unmute and you can ask the question. Jyoti ma'am. Jyoti ma'am.
Yeah, in chat box, one more question is there. That is, can we see the foliar morphology using Raman spectroscopy? Vijay Bhaskar, you can also.
Definitely, because already talked about 2D and 3D imaging.
Yeah.
When there is imaging possible in the nano size also, because Raman, where you are allowing the laser to fall on only a nanometer zone, 1 nanometer, 2 nanometer zone, you can expose the molecule. And definitely any molecule which is having its morphology, that can be 3-dimensionally viewable and 2-dimensionally also viewable. You can take the picture, you can take the image, like you do some sort of SEM image that in the morning speaker, I forgot his name, you came from US. He also showed some SEM pictures, AFM pictures, and TEM pictures like that. You can also visualize the particular structure of the molecules of plant tissues by using Raman spectroscopy. That's what in the beginning I told you, 2D, 3D imaging is possible. That's what it is promising. Spectroscopy is promising.
Yeah. Okay.
Thank you, sir. Vijay Bhaskar.
Thank you, Karthik.
Right.
All the way you have been with me and supporting technically.
Yeah.
Thank you,
Thank you, Dr. Upender. And dear participants, Dr. Raman Nageswarrao, myself, Dr. Upender, and Dr. Shashikala ma'am. And one more important person is here with us, who is visible on screen, Dr. Vijay Kumar. We all are appointed in University, Usman University, same time. And welcome, Dr. Vijay Kumar. And on behalf, we requested Dr. Vijay Kumar to propose vote of thanks on behalf of organizing committee and participants. Over to Dr. Vijay Kumar.
So thank you, sir. Vijay Bhaskar, sir. Thank you very much for giving me this an opportunity to propose a vote of thanks to our colleague, friend, and today's afternoon speaker, our young dynamic friend. So Dr. Upender has given a very important topic. And first of all, I congratulate Dr. Vijay Bhaskar Reddy, Dr. Raman Nageswarrao, and Dr. Shashikala for choosing Dr. G. Upender for this topic. So I hope that, and on behalf of all the participants, I can say that even though they are not familiar with the physics, but after his talk, most of the participants, they may forget their plant sciences and they may shift to the Raman spectroscopy because he has given this talk from top to bottom. I can say in simple word that, so if I ask to explain or describe a human being, we can explain from top to bottom, toe to head. So similarly, Dr. Upender has taken this topic from basic background. What is Raman spectroscopy? History of the Raman spectroscopy, and father of the Raman spectroscopy, and its applications, especially for one category, that is plant sciences. So he has given a wonderful differentiation between Raman spectroscopy and vibrational spectroscopy. So most of the chemists and physics, they may got some confusion. So which molecule give Raman spectroscopy and which molecule gives vibrational spectroscopy? But this topic is new for you people. But after his listening, I hope that you will clear your doubt. He has clearly mentioned especially two things. One is, how can we use Raman spectroscopy in plant sciences, especially for the determination of cellulose crystallinity? Sir, Upendra sir rightly mentioned that for materials chemistry, he can measure the determine the crystallinity by using X-ray diffraction. But as a plant biologist or plant science teacher, they don't know, or it is not possible to use the X-ray diffraction to measure the cellulose crystallinity. So it is a great and a wonderful technique to determine the crystallinity of the cellulose. And he has given a wonderful differentiation, that is by taking one homoatom and one another is heteroatom. How the bending, symmetric and asymmetric stretching is taking place, what is the dipole moment, and what is polarization. So if you're not clear in these two terms, it is very difficult to understand the Raman spectroscopy. The differentiation is very small between the dipole moment and polarizability. So mu is equal to alpha into A. It is a very important formula to understand the polarizability. And Sar has also mentioned the types of Raman spectroscopy, and especially he is very familiar with the SERs, surface enhanced Raman spectroscopy. And by using this technique, then how can we determine food, what we can say, quality? So I hope that this lecture will give the basic idea. Those who are not familiar with the Raman spectroscopy, this lecture will give an important basics to all the participants. I hope that all the participants enjoyed, because our coordinator, Dr. Vijay Bhaskar Reddy sir rightly mentioned. So almost 2 to, it is approximately 5 o'clock. Up to 5 o'clock, the strength, if you see the participants, it is 120. So hats off to all the participants for your kind and patient listening. So with this, I thank all the organizers and Karthik. Thank you very much for giving me this opportunity to propose a vote of thanks. Thank you, one and all.
Thank you, Vijay sir.
Thank you,
Joining
sir.
us. Joining us in the evening. Thank you.
Thank you, Dr. Vijay Kumar.
Thank you,
It's
Shashikala madam, for your
wonderful,
nice words about me. Thank you. Thanks.
wonderful words. And dear participants, please you all excuse our organizing committee team members, because every time we are starting every session on time, but we are unable to conclude it on time because of sessions they're going like that. And with your patient listening,
And indeed, we must be thankful to technology also, who made us connect. And of course, thanks to Hyderabad power supply. Today in the afternoon, power didn't go.
Over to Dr. Ramanji.
Yeah, thank you all the my friends. Dr. Vijay Kumar, Academic Coordinator, Osmania University, Nizam College, Osmania University. He's also Assistant Professor in Chemistry. Dr. Upendra, Dr. Shashikala Madam, Dr. Vijay Bhaskar Reddy. And finally, I'd like to make one or two these notices are for you. We are supplying every day Google Forms for the feedback and attendance sheet. Today also I have shared the Google that form in the feedback, in the chat box also I have shared, in all the WhatsApp groups also shared. Please go through it and please send the feedback form and attendance form. And there is no assignment today. Only feedback is there, broad feedback is there. You just fill the feedback and send it back immediately. It takes only 2 minutes time. And one more thing is, kindly bear with us. We are trying to include some of the questions every day. Today we have not included because the topics are a little new, a little different. That is why we have not included that. One more thing is assignments. Regarding the assignments, yesterday we have given the assignments. Majority participants have submitted their assignments. If anybody is not have submitted, please submit tomorrow. There is no time limitation for that. You can submit tomorrow also. So with these two notices or instructions, I'll hand over to Karthik for further concluding the session.
Yeah, thank you, sir. Thank you, everyone, and thank you for the participants.
Karthik, 1 minute. 1 minute. Yeah. See, on behalf of the organizing committee, I am telling since beginning. So all the PPTs available with us of the resource persons, we will be sharing with you without any that thing. If the resource person shares this PPT, then definitely we will share the same PPT with you in the WhatsApp group. So you need not to ask again and again. Definitely we are going to share all the PPTs available with us. Until unless it is a very specific research work, they will not share, research persons will not share the data. That will not be shared with you because that is we are not having that PPTs with us. Thank you.
Okay. Thank you, everyone. Thank you for participation. Vijay Bhaskar, sir. And I think tomorrow's session will start at 10 o'clock.
Yes, yes, 10 o'clock. It's a 10 o'clock.
10 o'clock.
Sharp.
So please come. So thank you, everyone. I'm ending the session, sir. Thank you. Bye.
Thank you. Bye.